sampling event for our 8th collection of the dive. This is a coral with a number of associates that our team is interested in. Yeah, and then it's really nice because if you want to frame grab and all the metadata associated with it, you just hit frame grab.
Yeah.
And yeah, that's
perfect.
I mean, I think probably get better at it, but I think it's very complicated with all the buttons and all the options right now. So, you know, some of the things like, you know, we tried to hit coral livery, we try to hit associates as much as possible. Some things are just...
Put that all in there. That's a generous sample. Very nice.
That one we should be able to identify right away.
Yeah.
But Narella is also one of the one that can be used for PopGen, so it's
yeah, no, it's good.
And got the crinoid in there too now. So you can write notes freeform just in this bar and hit submit, and it captures everything also. But you know, you don't have to fumble around with the buttons and just write it in there.
We'll go ahead and look. You can even look into camera.
Yeah, I can.
And
Oh, the crinoid's swimming out.
No.
So they do that.
It's okay.
tell me about.
They're beautiful to watch.
Is this the first time you've ever been on a ship like this? And what has it been like?
So kanabay naman ninyo, kayo nag-brain siya o experience ng kami na nangyay niya ng depot na tayo yung minawi kayo magdabay ay may pinabay spongman tayo ninyo ng nila depot the expedition yung antik pangati research yung arawat in the marine ayun kayo spongman tayo ng brain kawan ang may sabaya kayo marine na nantira na ito tayo nagkakain tayo yung ispaya yung kayo man tayo in the Seamount kaya lagay tayo
good?
We're good.
All right. Hi, Cody. Hi, Jared.
Hello.
So you guys know we're just gonna keep trying to traverse the line, but we're probably gonna see a lot that we want to
No problem.
zoom on.
Let
So
us know and we'll do it.
there was something down just right below us. There were these pink corals, these smaller pink corals. Did you see those? Can we take a look at one of those?
Do you want me to send it?
And hi, everybody. My name is Alexis. I am a research scientist with the USGS. And I'm out here as a guest investigator with Woods Hole. And joining me on our watch is...
Good morning, good afternoon, everyone. I'm Brian Kennedy. I'm a PhD candidate.
Yes, sir. Yep. We are at a seamount in the high seas, or what is known as an area beyond national jurisdiction. So no country owns these waters that the seamount resides in. And we have been working our way up. I believe we started at about 2,200 meters, and we'll hopefully get up to around, what's our end?
About 2,000, I think, was the...
Oh, we're at two. We're only we're
Okay.
2030, so a little ways up, and looking at the diversity of corals and sponges, the types of geology that we see, and then also the animals that are associated with the corals and the sponges, other types of invertebrates, and then any fish if we see them, but we haven't seen a whole lot so far.
I saw one or two rat tails earlier, but I haven't seen any recently.
Okay. And we just spent a good amount of time imaging this large primnoid octocoral to the left, and we're going to position to be able to look at these pink corals in front of us that are likely hemicorallium, so a type of precious coral. Early, we are seeing these live ones for the first time so far today.
We haven't zoomed on hemicorallium yet.
Yeah, that's what I've asked to do. I love these little kickouts that we just have now. They're so
It's all right.
Used to working with Phoenix Islands is starting to post stuff on Facebook.
Cool. Work with magic. So what do I do with that clip? On YouTube again?
Yeah, that'd be great.
I'm gonna do that now.
Yeah, whatever.
Okay.
All right. Awesome. Thank you.
Next couple hours.
I'm gonna learn how to use this camera again slowly.
Is that?
Alexis, you want me to mess with the iris and ISO stuff?
Totally fine.
Awesome. Thank you,
I mean,
Cody.
I'm a little, I need to get some walking stuff going.
Yeah, yeah.
Let's see here.
It's overwhelming. It's good
Okay.
in this room.
She's a hemicorallium Pacific. I think it's one of two species. So.
Steve, do you have comments on what type of hemicorallium this is?
It's one of two species, but I'm not
Okay.
sure. But we can identify. We've collected a lot of this
Okay.
type from many cruises, so I'm pretty sure we can find a good idea.
So we've collected this, you can't hear Steve in the back of the room, we've collected this coral before on previous cruises, and not this seamount, but similar seamounts in the region, and so likely just from the video we'll be able to identify it, so that does not warrant us collecting a sample, but it's good to get the up-close zoom so that we can really look at the polyps and the branching structure.
Which is where? Okay. I just want to make sure I wasn't...
Yeah, great.
You can write in any number of things.
Yeah.
All right, thanks guys, we can
Easily
move
searchable.
on. Look at those little wispy sponges up there that look like cattails.
Yeah.
Do you know what those are?
No.
I know sponge, that's about, yeah, carnivorous sponges. Yeah, sorry, do you want me to tell you things as we?
I'll ask if
Okay.
I need the
Okay.
clarification. We apparently missed corallidae in the.
Is
radio
hemicorallium
buttons.
on there?
No.
Really?
Both of them are somehow
I don't believe
missed.
you. I made this list. Oh yeah, there's some bigger ones out front too. Beautiful. This is probably a similar type of primnoid to what we saw earlier. So we're not going to spend a whole lot of time on it, but if you guys want to just like get yourself nice beauty shots kind of flying by that, that's awesome.
Do you want to take some more water samples? I think we've only done the one.
Yeah. Okay. Thank you.
Do you see it in here anywhere?
No, you're right. We'll add it next time. So while we're out here, we obviously are collecting all of this video imagery that goes back to the various labs in order to analyze it and confirm IDs for corals and sponges. But we also will take samples along the way, so collect certain corals, sponges, and associates so that we can bring them back if they need really up close diagnostic features for identification. But while we're on board, Brian and I are sitting here together. I am working with the ROV pilots to help us decide where we're going, and then Brian is logging, so he's sitting at a computer and documenting what we see as we go so that we have a reference and essentially a nice long data sheet when we end each dive, telling us what we saw, where we saw these. Let's go up to more of those pink hemicoralliums that we were seeing, but straight ahead there's something that looks a little more interesting to me, and that is a very large colony of probably a different type of coral that we haven't seen yet, or a very large hemicorallium. So the hemicorallium is a genus, but they are within the Corallidae family of corals, and they have they're called precious corals because their skeletons within the family, there's different colors of this, either really beautiful pink or red, sometimes white skeleton that for even to this day are sometimes harvested for jewelry and things like that. So they are a protected group of corals, and that you're not supposed to take them for commercial use. But they are very slow growing, and they have these really delicate polyps that help you to identify what type of coral they are.
Can we land out and get a good view of this one?
Yeah, why don't we? Yeah, so even within this one little boulder right here, we have at least 3 different species of coral. It looks like a couple probably dead sponges at this point. Those kind of greenish gray colored sponges looks like they have a lot of sediment buildup on them, so over time as the water is passing by the corals, that's how they get their food. They eat particles out of the water, but also if there's a lot of sediment in that water, it can get caught up in their tissues and essentially ends up suffocating the sponge.
It looks like, is that a black coral over on the
Yeah.
right side there?
Yeah, so Brian's Litharian, and those corals are called a black coral for a common name because they have a black skeleton underneath their tissue. Oh, we're good? Okay. Yeah, so another really large colony, just like some of the other octocorals we've been seeing. So the lasers on the screen, which are green, kind of right in the center of the screen, are 10 centimeters apart. So it gives you a bit of a scale for how big the overall colony might be. So we're looking at meters. And there are at least a couple associates on here. So Tim Shank is on board, who is interested in the coral associates. So we have a shrimp up on top.
Yeah, we've got a second one off to the left of the frame right now.
This little guy.
Yeah.
Yeah.
And then is that
Maybe another shrimp.
solitary hydroid or something?
Let's see how close we can get into there.
Or are we looking at the
Or
back
just
side
like
of an
a,
anemone?
yeah, maybe. Or like some, but there's also an anemone just to the
Oh,
right.
no, I think we're looking at the, oh no, I think we've got a little crawling amphipod here on the bottom right as well.
So we're on the opposite. So obviously you can see all the polyps are facing on the particles more easily that way. But there's also a relationship between a certain type of worm that will burrow into the skeleton of these corals and makes them actually quite fragile. But you sometimes can actually essentially diagnose the species based on the type of worm that you find inside.
And then what is this white one that just came into the frame here?
So this is somebody that has been colonized by some type of zoanthids.
Oh, yeah, you're
And
right.
we have, I think, a homalid crab up there, actually. Let's see. I'm sorry. I'll get my zoom and everything down eventually. Yeah, so there's a base of a previous type of coral, and then growing over it, all those larger polyps are zoanthids, so a different type of cnidarian that essentially colonizes either dead skeleton or live skeleton and takes over that coral to make it its own home. And then we have, it looks like 3 different squat lobsters living on that colony as well. And at the top, that kind of bushily, brambly bit is hydroids. So another associate. I'm trying. Give me a minute.
Hey Alexis,
Yes,
is
ma'am.
that black coral actually attached to the bottom?
What?
Or is it attached to the hydroids?
That's as much I can. That's a, I.
Okay.
Yeah.
Fine. Okay. Right. Thanks.
Is that good for you?
Hold it a second.
And actually attached to the bottom, or is it
Branch tips.
attached to the branch tips?
What anthropitherium?
That is a. I think that's a hydroid. Am I wrong?
Sorry, is that hydroid? That's what I mean. Sorry, is the hydroid attached to the?
I think so. I can't
Yeah,
pan
looks-
up anymore,
looks
so
like it.
Right. Great.
pull down at the base. Did you see that?
Yep.
Okay.
Wow. Really nice.
Huh?
Very nice.
Good.
The Mathapathes has got something on it too, doesn't it?
Does it?
Yeah, you're right.
That really bally, bushy one.
And you can kind of, there's a very hard to see siphonophore floating through,
Oh.
just kind of coming into center frame right there. It's very thin and gentle. I've lost it now.
I was gonna say I can't see it. So.
You'll probably see it again here. It turned invisible. I lost it.
There's a crab. Tim, do you see that
Yeah.
crab on the dead sponge? That all white
No.
right here.
Oh yeah. Yeah. There's something like Mathapathes too. Quite a few corals here. That chrysa looks pretty big bush.
Yeah. Do you want to look at it, or you want us to keep going? Do you want to look at it, or you want to keep going?
Did you fire one this
Can you look at the chrysa gorge? Is it
Okay.
We
same
have
time?
not.
I'm gonna look at this
Let's look at
little
that.
coral over here in the corner. Can I just take a quick look at it?
Okay, is it that one right there?
The one that's kind of bushy up in the
Oh, okay.
Yeah.
That's the chrysa bushy. Maybe you want to pull that bushy.
I'm done. We got one. Can't get any more. Can't handle it. Great money.
Let's see what this is.
Oh, it's very plainer. It's not as bushy as we.
The backside?
Yeah.
Cattails.
Cattails.
Can you guys take a look at the cattails in the back?
Yeah, they're probably polyderma sponges. Look at the crinoid just falling
Yeah,
through,
yeah.
falling through the water column over on the right.
It's so hard to really look at everything, but you also got
Yuki snacks. Remember, that's a nice little forest up there.
And it looks like a bunch of. Those are all hydroids up at the top. They look
On
like.
the right, yeah.
Yeah.
Those little tufts of hydroids.
See, that's crazy.
When we get done with this chrysogorgia, I would like to
Okay.
go over and take a look at the stalk sponges.
Sounds good.
That's I'm telling you, it's a
different sponges.
That or they could be those hydroids. They're kind of yeah.
there is a lot here.
But this also goes into the observation we made that there is a 2,000 meter, probably 2,000 meter diversity maximum, and then it kind of goes down as you go up
Okay. Here we go.
from what I've seen on the
Cool.
course. Diversity in life is the gorge yet.
Oh yeah, there's a little squat lobster in the middle of there.
Which is, yeah, and that's exactly what we saw. We saw hundreds of them. So in a way, this is actually more reminiscent of our feeding sites than, say, the Line Islands, which is kind of strange because
Tim, you see this?
Yeah, I got it on it. You full zoom?
Yeah. Oh nope.
Your plate.
it's a biplanar colony. It's not bush. It's not a.
No.
There we go.
It's
No,
biplanar.
you went to Bush, didn't it?
It's just hanging out in there.
Two planes.
Just chilling.
Just chilling.
Chilling like a villain.
I mean, it's funny. You always find these same. Tim, you can comment on this too. You always find these same types of crustaceans living in these corals, but you don't see them like just living on the rocks.
Right.
Which is just bonkers, right? Like how. Look at this like little wispy cattail.
Okay, I'm walking around.
Like when you do close zooms, can you frame grab button?
Oh, what do you want?
Just hit the frame grab
Hit
button.
rainbow when you get something like this. Yeah.
Okay. Okay.
Yeah.
You can never have too many frame grabs.
Well, I mean, we're taking them automatically every 1 and 5 seconds.
It's true. Truce. Every 5 seconds.
Yeah, there's little and
Yeah. So there.
But yeah, I can certainly take more.
but that gives us the metadata associated with it,
Well,
environmental data.
I mean, that's all pulled in the, I mean, frame, C-log is taking a 1 second frame grab, that's
That's-
second associated with all the aux data too.
Is it full resolution?
Yeah.
I think it is.
Yeah.
It's just that you don't have, like frame grab gives you a column at the end where like all of those you can sort them out as frame grabs. So like if there
Yeah.
are very specific things that you want.
Yeah.
So one of the things that always amazes me in the deep sea is the patchiness. Like we look at this rock and we have a good sized rock boulder here, and we see just this one little corner that we're trying to image in, and or you've got a couple different hydroids here, and then the cluster of sponges that's just outside a frame. It's one of the things that always fascinates me is why is that one spot over there got all the sponges and the rest of the rock has none? And when you think about a lot of these are some kind of broadcast spawners or things that it doesn't quite make sense to me on why these things are so isolated in some areas and not others.
Yeah, so this is a hydroid, which has a little crinoid on it as well as, looks like a couple barnacles, which we won't spend too long on. If we can, Did we have hydroid as an option?
and
'Cause
non-associated
we should
inverts.
probably make a.
It's nice
we-
in here.
It is okay.
Yeah,
I was gonna
hydroid's
say
in
we
here.
should make a list of the things that I forgot to put in there. Here's
So
your.
these are a type of sponge. They're these are probably something similar in the Piloderma genus, is my guess.
They're carnivorous or no?
No, I don't think
Okay.
these are. No, the cladorizes or carnivorous sponges a lot of times have little short barbel type things sticking off the sides.
Oh, okay. We have a number of crinoids, but nobody else hanging out on there, which is kind of interesting.
And this is a great example of why we talk about sponges and corals in the deep sea being ecosystem engineers. They're creating new habitat where these crinoids and whatnot can climb up to and get out into the current. And you can see there's a decent amount of movement here.
Yeah.
Some of that could be our thruster wash, but we're pretty good little ways
No,
away
I
from these
think-
guys.
yeah,
We're
I
pretty
don't
zoomed
think-
in, so it's probably all natural current as we climb up this side of the seamount.
Yeah, and it's interesting to see the different sizes of the sponges too. So some will have been there for a longer amount of time. Have these patchy areas, but some of them, like it almost seems like there's pulses of recruitment, right? Like they all recruit at the same time.
But only in that like 3 square
Right.
meter area.
They find somewhere that they really love and it works out. But what could have happened is that you had really small recruits right next door and they didn't do as well.
Right.
So before they ever actually established into anything, they wiped out. Okay. Good.
All right, so we make some tracks.
Yeah, I know. Yes, we should. It's a very delicate balance of wanting to look at everything and actually moving the ROV. So we have some, that white kind of brambly stick looking is probably a dead hemicorallium. So those pink corals, when they lose their tissue, the skeleton is just white and hangs out there.
No, like I said, that might be a Bersingid sea star, kind of on the top right there. I think that's the first one I remember seeing.
Or it might be a crinoid, actually.
Could be.
Yeah,
But
one of the two.
Those are long arms for. Let's see if it swims here.
Yeah, I think you might be right. It looks like a Bersingid. I think a sea star would have jumped ship.
A feather star, you mean?
Yes.
Yeah.
It's day one. I shouldn't be making those mistakes yet.
It's day one and it's only 4. It's 4 o'clock in the afternoon, not morning.
This is very crusty stuff.
You want a fire water?
Yes,
Let's get a
yes.
water bottle.
Like you can
Do we want to? Yeah, why don't we get it around all of this?
see this is not rock.
This.
She think would be good for an RV getting
Thanks, Cody. So we're firing a Niskin, which is a bottle mounted on the ROV that can collect seawater. There's an Anthomastus right there.
Oh yeah, isn't that on?
And we use the water for a couple different things. So we have a collaborator that looks at eDNA, so environmental DNA. So you filter the water and then you take that filter home with all of the particles on it and you can sequence for DNA and see if you can detect certain organisms. So having that water sample partnered with the video where we know what is there can help us to essentially fact check what the eDNA is saying. And then we also use that water sample to look at the microbial community. So Anna on board does more microbial biology, and she'll do, are they just doing like metagenomics of what the water is?
or specific primer sets.
Okay, yeah. So just looking for what microbes are much diversity or not as much density of other animals.
All right, and that was sample 9. Is that
Oh, did it fire?
We're checking, but will be sample what?
It fired what number?
Number 1.
Thank you. Number 1
Let's get one.
and sample number. What do we, Steve? What sample number did we leave off on?
That was 9.
9. Yeah, and if we can just kind of pan around that feature.
Yeah, 1 second.
Cool.
Just want to verify that it did
Uh-huh.
fully fire.
Okay,
Good.
great.
Good to go.
I love those new to me buttons
What's
for
that?
pushing the NIST
Yeah,
ones.
yeah, they're awesome, right?
Yeah, versus having to pull something back
Yeah.
into.
They get the job done.
Yeah, it's great. Yeah, so we'll just kind of go around this feature and then head on our track a bit more. So you could just do like a frame grab or something to.
Yep.
Yeah, that feels. It's a bunch of hemicorallium. It's like all over the place.
Yeah, look how thick the base is on that.
What'd you say?
Just how thick the base is on that bottom one.
Yeah. Well, that's the other. It's like a. And this is also like a balancing act. You know, what at some point we find those broken skeletons likely because they get so heavy after living there for so long that they just fall from gravity, even though it's in water.
Did they get a transect yet?
No, because Steve didn't want to do it.
This is steep, yeah.
Yeah. Glass, a couple different types of
Actually,
sponges.
Look at how that one, all the arms of that one, but the stalk
Yeah.
are broken off.
Yeah.
Yeah. I think so. It's slightly far away. This is very.
And like you can, it's interesting. There must be like
In the northern line island, which
some kind of current situation. There's so many right on
Yeah.
that face of that rock.
really overhangs. It looks trapped there.
Yeah. Look at it. It's like a almost a semicircle right over that.
And I think further south.
Oh yeah.
That's
That's cool.
But I think quite geographically it's in the scans from the Hawaiian group. But when you move further south, you get more influence from the Western Pacific and the Southern Pacific. New Zealand especially.
Yeah, that is not like a good place to put the RV.
From corals, the equatorial influence.
So that's a good thing for everyone watching at home to think about is like while we're here talking about the science and looking at the thing, we've got the group of super talented RV pilots here that are not only enabling our science but trying to keep the vehicle safe at the same time. So they're judging our scientific request with making sure that the vehicle is safe at all times. And so you see those little box canyons like that because you got to remember we've got a tether all the way up to the surface that allows us to control and power the ROV that can get hooked in those kind of things depending on how we're moving around. It's got floats on it, so it's floating around. So there's a lot of, complicated, it's a complicated ballet down here to get us to see the one video stream you all are seeing, and we have a whole wall of monitors and different perspective cameras the pilots are using to be able to safely navigate thing and point out things that we're not necessarily seeing because we're staring at our science cam almost exclusively and ignoring all the situational awareness cams around that the pilots are all bouncing their eyes across. So they help us a lot sometimes finding other things as well.
Yeah, I always end up looking at the pilot's cam in
Yeah.
addition to mine because I find that I see things
See
more
what we're
quickly.
seeing,
Yeah,
yeah.
yeah. Can we take a quick look at this little coral right here? And then, yeah, so there's looks like the long kind of Y-shaped coral is probably a bamboo coral.
Looks like we've got one associate kind of down on the
Oh yeah,
front
hanging
base.
out on the base. Or like the... And then I think behind that will be also a new
Yeah, I don't
coral
know.
for us today.
I'm not sure what that is yet.
I have ideas, but I like to get closer
That's
before
probably
I.
wise.
Yeah.
It's unusual how common that is. Maybe it's not. Maybe it is just like a very late life branch.
I don't think we haven't seen many bamboo corals today either yet, which is kind
Yeah,
of
it's
surprising.
been a little bamboo light.
Bamboo light.
That's spiny armed.
Yeah, I think so.
I think so. Which makes it.
Ophiocanthidae is a spiny-armed brittle star.
I don't know if we ever used those.
It looks like too that the tissue is missing from where that. We'll see when we take a look at it.
Great.
Yeah, so we say it's a bamboo coral, similar to black corals because of the way the skeleton looks, and so these bamboo corals have essentially what looks like bamboo. You have white nodes and you have those darker inner nodes. And then, interesting, Tim, we have a
Yeah.
Ophiocanthid,
Yeah.
but it also looks like the tissue is missing from where it is.
It looks like is that recolonization lower or?
I would think it's the original
Okay.
coral, but
The polyps
actually
just look
no, you're
smaller to
right.
me.
Yeah, you're right.
I've noticed that on other cruises too.
Oops.
Master schema on. That's gotta be.
I'm coming back to that one. There we go. Yeah, looks like maybe like a stolen, like another type of octocoral. Not the big bamboo polyps that you see.
Do you want to collect it? It's a stolen Earth coral.
No, that's too deep, like that's way too far down the base. This thing is tall.
Oh.
Is that really? That's really bubblegum over there now?
Yes. Certain.
Yeah, so look at the polyps up at the top. Have that really cylindrical, like
Yeah, they've
barrel
got the
shape,
little, and then a little
and then
bulbous
these
top
little,
and these
yeah.
little things
So
down.
interesting. Okay.
That's cool.
And then we're gonna just try and look at this guy that's behind here without having to
I saw that.
move.
I wasn't sure if that was a leg or something or. Oh,
Let's go in as much as we can. Yeah.
it's got stuff on it.
Steve is Paragorgia. Okay. So it's a Paragorgia, which is a different family of octocoral than we've seen. And they have zoanthids. So the pink part is the Paragorgia, and then the zoanthids are another type of coral that will colonize other coral bases, and they don't actually create their own skeleton. They just take over a different colony. And then an astroschema.
Yep.
Okay, an astroschema in the middle is that brittle star with those really wormy, like, legs. Wormy like legs.
Quote of the day, Alexis.
I'm pretty good at those.
Do you want to keep the list of things we're missing and see? Like, we don't have zoanthid as an associate.
Okay, good to
We
know.
don't have
Yes, I will keep the list. Yeah, we can head
Move on?
on.
Okay.
Awesome. Thank you.
Sure, I hope you're gonna sample that.
You'll get your chance.
So, why was some of the tissue going over that bamboo? Could it have been a sea panda?
It could have been a sea panda, or it could have been another type of predator, like a snail, maybe.
Panda snail?
Yeah.
Did you guys hit the coralivore button?
We didn't because I don't know that that's.
Okay.
I don't think that Ophiocanthid is doing that. I think it was missing tissue.
Oh, fair enough.
'Cause it's not
Beforehand.
really-
Yeah, I agree. Okay, so while you're traversing, I want to take a minute to acknowledge the fact that this is the first dive of the first cruise of Challenger 1 as part of the new UN Decade of Ocean Science. And we are so incredibly excited to be a part of this and to kick off this dive. And on the day that it was announced, on World Ocean Day, on an AVNJ Seamount. So, you know, all in all, a pretty good day. Not to mention all the pretty amazing sea life right in front of us. But we're really excited for all the ocean actions that were UN ocean actions that were endorsed today. I think it's a really good sign for of the decade to come, and all the amazing work that's going to happen. So congratulations to Challenger 150 and all of the other fully endorsed UN Ocean Actions.
Yeah. So Challenger 150 is essentially a
It's an initiative and a collaboration to try to help standardize some of the deep sea data and make sure that it's publicly accessible, but also, you know, there's to help to enable some cross comparisons across multiple sites and to try to really enable that. There's a lot of different ships and a lot of different scientists who all collect data in slightly different ways, and so sometimes that standardization can be really helpful when you're trying to take a bigger look at the whole ocean.
Do you want to take a
Yeah, let's
look
take
at
a look at that.
Yeah, that kind of conglomerate. And the other thing about Challenger 150 is that it is an effort to really diversify deep sea science, so bring in and include people from nations that have not historically had a lot of deep sea exploration resources in places that are their own waters. So we're in international waters, but at the same time, there's a number of countries that have a lot of deep sea space within their EEZ that have not had the chance to actually do the exploration to find out what's there.
And so we're so lucky to have Anamare Tanaba on board with us from the Republic of Kiribati. And she just got off watch, but you'll hear from her a little bit later. And it's pretty great to be able to, actually there's a whole lot of nationalities on board this vessel right now. I don't actually know the total count, but it's pretty great to have such an international group in international waters.
And some people in the chat are asking about like the brittle stars and if they eat corals. To a lot of us that have been doing this, and Tim, who studies the associates specifically, that's not really a common occurrence that we see. We see them associated a lot where they're living on the coral, but it's never really seems to be that they're actually eating the coral tissue. So that's where earlier when we saw that kind of blank tissue, but the ophroid on there, I would assume that that tissue necrosis happened from something else, and then that brittle star is just kind of hanging out.
Well, this is beautiful.
Yeah, so a lot of
Pleurogorgia.
which is this very wispy chrysogorgid, so one of the gold corals. And they definitely like this spot a lot. We've seen in a lot of these, and Steve talked about it before, that we often see kind of pulses of these where you have hundreds of them in one spot. I'll analyze this video later because that's going to take a long time to count all those guys.
Associates on this.
Yeah, right. Not any associates really on this type of coral.
It's interesting, you see this big pulse and high density community of the Pleurogorgia here, but the sponges here, for the most part, appear to all be dead. Except for this one over here.
Stalked guy.
Yeah, but you've
you're
got
right, yeah.
3 or 4 dead sponges with this one stalk sponge over here. So it's almost like some of the conditions changed here, made it unfavorable for the sponges and more favorable for a recruitment event for the corals.
Yeah, and we've said before that, like, especially with sponges, it would be very hard to figure out how long those have been dead. It could be for.
What is that right there? Is that a crab?
You want to look at
Can
that
we zoom-
crab?
Can we zoom in?
Can I zoom in?
Go ahead.
Okay.
Sorry.
Pleurogorgia genus within Chryso, or is it its own, or not Chryso within Para?
It's in the chrysogorgid.
It's in Chryso.
It's a chrysogorgid family.
Okay.
Yeah. Are you good, Tim?
Yeah, I'm fine. It's a gallopy, essentially. It's on the rock.
Yeah, it's on the rock. Cool.
Good?
Yeah. We can keep going.
Tell me if I'm bugging you behind.
Oh no, I can't. You're fine. Are these so far the buttons that we're missing, Brian?
That's all I know of,
Okay.
yeah.
We're missing an amplify button.
Oh, for what?
Well, I thought we
Associate?
were specifically not doing an amplified budget for associates.
Yes. Gotcha.
Can I just spell that again? Zoom.
Yep.
Yeah, so the major difference is the chrysogorgids all have these plate-like scories, but actual Pluralgorgia family was broken out by
Oh
something.
yeah, that's much better.
I don't know what the distribution is on it right now.
To get to.
This Pluralgorgia
I
still
think we'll probably go
embraces
to
the
midnight,
predate family.
Unless it's terrible.
Yeah, because I mean we've barely moved since we've been. Oh, look at that core livery.
Well, there we go. Hit the
what?
core livery button.
Not yet.
What?
No, okay.
No,
So
It's gonna shatter. That's
we do you want a good
I just
image
want a
of
good
it?
image of
Okay,
it.
we'd like to image the sea star.
We have my basket's full. I have more than I can process tonight.
Oh, okay.
That's an interesting point. Maybe it's the ones that are precarious like that are.
So we have a we all are yelling core livery because one of the goals of this cruise and the dives on this cruise is to document instances. Steve, do you know what kind of star that is?
No, right now.
Okay.
It's very
So look at how much of it it's eaten
Look
though.
at the impact of the,
Yeah.
Wow.
right?
Yeah,
Get
so
a good image of that.
we want to
I'm
get,
gonna let him do that. He's got it.
if we can, let's get the most planar image straight on. I mean, we're basically there. But
Yeah, Jared's doing great.
This
Yeah.
is fantastic. Can you?
I'm gonna take down the ISO just a little bit.
Okay,
It might go really dark first.
that's good.
That's beautiful.
great.
You like that?
Yeah. There's a little oaf there in the middle too. You see that?
An oaf. That's a crinoid
And zoanthids.
and zoanthids.
Look at that coralivore.
I've never seen it like that. I mean, this is like it's hanging off. He's gonna have to work his way all the way
Can
back
you,
down
take a couple of
before he can
screen
eat
grabs
more
for
of
me?
that. That's a great image. That's
It is great.
very good.
You know what's funny? I did my PhD on coralivore in shallow waters,
Yeah, beauty.
and
Yeah.
when I started studying it, everyone I was with was like, "I don't see coralivores very often, but once you look for
Well,
it,
you're
they're
right.
everywhere."
You're-
And it's going to be the same here, I think.
I send all my stuff to Mom.
Look at that!
Yeah.
Wowee, that is. You know what that is? That's like the squirrel at the end of the branch. Where you're sure it's going to fall.
But that's also what's so interesting to me is he's at the end, right? Or is gonna have to work its way back.
because what am I seeing here? This area here looks
So
like
that
the
is
polyps are-
zoanthids.
So yeah, so that's zoanthid
yes.
regrowth already on
Yes.
the predated area.
What do you
Yes.
see?
And then
What?
that
What do you
is
see?
We see a very precariously perched coralivore, which is highly entertaining and beautiful to look at, but it's also a prime example of how much
And
some
look
of these
at how-
coralivores
yeah.
can decimate
Look how
a
the
coral,
2
and then how
feet
quickly,
are
I'm assuming
grabbing
you don't have
onto
to at
it.
the time, but how many other
Look how
organisms
the
can
2
use
feet
that
there
space?
is
Oh, yeah,
extended
just out.
out in the middle of nowhere.
These corals, we always call them, ecosystem engineers because they're building the foundation. Lots of other organisms use them. There's tons of associates, but look at the number of associates on the dead coral space as much as the live, right? And they're clearly using this structure. And now we're just taking bets over when the sea star is going to fall off.
We can speed it up for you.
no. Are you good, Randy?
Yeah, I'm done.
Okay.
That's awesome.
That was great.
That was really great. Thank you.
Thanks, Jared.
I love coralivore. I mean, I don't know. It depends who you're rooting for. The coralivore or the coral.
Yeah.
But right now, I just like the interaction. It really is everywhere. You're going to see it everywhere now.
That was a very good instance of it.
That was
for-
perfect.
Yeah.
If you can't,
Yes.
Wait.
come around and try to do this snap zoom on the back of the
On what?
back of the zoom on the predator.
Oh, you want to
He
flip
wants to-
around?
he wants to
You want to see
ID
the
it.
front?
We can see
dorsal
the.
side.
Okay. Okay. So if we can
We're not
see
quite
the
done
front
yet.
of the. That's actually
So
yes,
we can tell you what it
please.
is. If you look on your, got your sheet?
Oh yeah. Let me find my sheet that you made me.
Yeah, but look at that. Thought we were done. Now we get to look at it from the other
Well,
side.
Tim's right too.
Yeah.
That we should look at.
Absolutely.
Here, Eva Plasoma or Mysterious?
I don't know. Eva Plasoma?
That's what you guys collected in 2017. I don't know. I don't think it's that. It's just that. But it's not.
Let's see how puffy it is. Looks like it's in Mysterious.
Do some frame grabs or beauty, whatever you want to do.
I'm working on it.
We'll get some professional help.
I'm honestly sure that there will be a screenshot of this on Twitter very quickly, and Chris Ma will quickly.
Are you fully zoomed?
Jared's
Yeah.
controlling because he's flying, so I'm not gonna.
That's cool. That's excellent.
I think this is great.
It also really speaks to how strong these skeletons are, right?
Yeah,
I mean,
actually.
That's a pretty heavy.
That's really surprising to me because you guys know from sampling some of these, they can be really fragile. So it's surprising to have such a heavy organism sitting on that tiny little branch.
So what's weird
Well,
is to have the crinoids on live parts too. That's really unusual.
I mean, the question though is mass versus buoyancy weight. So
Oh.
Pretty sure I'll be fine.
there might be a fair amount of, it might be heavy outside of water, but I wouldn't be surprised if these things are very close to
Oh, that's a
water
good
density.
point.
So they're probably not having that much drag down.
And
the
it's
board.
very spread out, right? So it's
Yeah.
spreading its weight as much as it can across that surface. I think that we're good. Yeah, Tim?
Yeah, so you got a snap on the opioid. That's what I was hoping for.
Oh,
We don't
You're
have a
right
snap
in, you're right in
rate.
the point.
They're right in.
You're all the way in. It's good.
So you look
Good?
we're good.
That is a
Thank
great
you.
shot.
Well, I don't know. When it's baggy like that.
If Mysterious is all has larger spines coming off.
We have a prim note up there that we've ignored, but it's also important. We don't need to zoom on it, just making a comment.
Oh, look at
Yeah,
the,
how it's
this,
growing
yeah,
within the
look at
rock.
how
But
that
I
one
wonder
has
if that
grown
was
the
eaten.
shape of the
You
rock.
See? Half eaten.
Oh,
Right?
it's also like a planer. I love how it's growing in that
That-
space,
yeah, that's
though.
really
That's
cool.
amazing.
Can we zoom in on that a little?
Absolutely.
Just look.
Like in this, I never understood that. I've seen that all over the years.
Well, I think
I bet that was half eaten.
So look, I think there is water. There is more gap space in there than I think we are aware of. There's got to be water flowing through that, like coming
Yeah.
in the top and coming up the other side.
I guess you don't know what you're saying.
No, there could be a current through there.
But you don't often see corals don't have pink.
Wow, yeah, just taking up all that space. I feel like that's unusual.
Is that zoanthan?
Yeah, zoanthids. That seems to be almost all of them have some level of zoanthid
Makes me think
colonization.
it's paragorgia, but
It's not.
not. Oh, it's little.
So you do have an associate on the.
Yep. An astroschema, it looks like. Smoother arm, brittle star.
Yeah.
I think we're good.
Yep.
Good?
But I'm just gonna talk. Is that white skeletal base would be one of these pink chemicoralliums that's all the bits have broken off, and that base is just left.
That was really good.
It was like great for imaging too, with the
It
black
was
background. You can't get
beautiful.
better
Nice
than
job,
that.
guys, for the imaging.
Gorgeous. New cover shot for the cruise.
Yeah, honestly. It's gonna be the new Victor Gorgia.
We need a new Victor Gorgia. That's gotten a lot of play.
It has.
So now you're gonna make tracks?
Now we're gonna make tracks, I
Did you
promise.
say that?
I know.
Bam.
Oh, let me write that down.
I don't know. This landscape is really pretty, though. But yes, make tracks.
I know, we did make some ground, so. But I feel like anytime we say that, then we find something really great, which is also good.
Fine, just
right?
park and do nothing.
No, I would never.
I would. Just watch. No, I think there's value of just watching the ecosystem. It's something we're always moving.
I
And,
mean, that's true.
The way we explore right now is we land, we take a look, we move function.
Yes.
These high density coral gardens, we document and move on. When I think about scuba diving, when you go out and dive on the reef and move across the reef quickly, you see all the things, but if you just sit sometimes for spend an entire hour of a dive sitting and looking at one coral head, the patterns, the interactions, the different species that come out when the 10,000 pound giant loud bright thing has moved on might be very different. And
You're
so
right.
it's something we don't do a whole lot of in deep sea science right now.
We have
Although-
to think about why, or the why is valid too, and that we have limited resources
Oh,
that
absolutely.
can do this type of exploration. And time. And there are cable to raise and things where there are videos happening on the bottom of the seafloor. Can we get a quick zoom on that yellow coral coming out?
Rock looks pretty cool right there. That's really puzzled.
A plexard with the
Oak in the
astroschema. Yep.
All right, what makes these things? I've wondered about this forever. These little 3-point connection mucus nets.
I don't know what makes those. That's
Nope.
great.
Don't know.
Good?
Yeah, thanks.
It's got to be some kind of worm or something that's just casting a mucus net for
Yeah.
food capture, but they're everywhere.
That's the deepest one for sure. That's what is
There's a crinoid hanging out under there, it looks like. And some, maybe a metallogorgia, maybe?
I think it's one of those weird. I think it's one of those like the same chrysogorgid we saw before. Don't you think? The pink one? Yeah.
Looks like hemicrime at the top, but I'm not sure about what we have down below. So we'll take a closer look.
Is that an Anthomastus?
It is. Good.
All right.
I didn't see that.
It takes 2
Yeah.
or 3
Or 4.
or 5
Or 5. As many as we can have.
or the whole team. Best team in the world.
Yeah.
Yeah, look at that. That's cool. There's an associate in the middle of that too. I didn't see what it was, but something.
What's that?
Well, this is something that's been colonized by zoanthids,
Okay.
and then it has an astroschema on it. I don't know what the original coral would have been because it's completely colonized. And then we can look at the one that's right below it too.
And that's not, first of all, we're not permitted to do anything like that. Like,
Some kind of.
So it's a chrysogorgid. I don't think. Would you say this is? I would not say this is metallogorgia.
No, this is crazy.
It's just okay. So this is similar to the one we got earlier.
What's in the middle?
A chirostylid.
Is it? What's the deal?
The crab,
Yeah.
or a Europtychus is what Tim's calling them.
All right.
Yeah,
Europe tigers? Okay, no.
Europe. Yeah, Europtychus. But it's also a chirostylid, so they both work. That's great. Thank you.
I mean, this is me talking, so.
I know.
Fair,
Yeah.
but-
True enough.
We just passed 2,000 meters over there.
We've just passed 2,000 meters. Well, yeah.
We're only going to like. We're not. It's,
That's a cool. Look at that.
No,
Woo.
the rock here has been like it's felt like it's been puzzle pieced together the whole way. There's all these little things jammed on either
Fractures
side.
and stuff.
Well,
Yeah.
I think it's the opposite of puzzle piece.
Yeah, it's fractured
It's
apart.
fractured apart.
Well, fractured, but you see these fractures where but it's wedged perfect. It's kind of amazing.
Exorbitate species too.
Don't want to work?
So,
So we started, Randy. Did we start at 2,300
I can tell you.
from here?
Yeah. Well, I know here. 2259, and we're only going to. We're basically sticking around the same depth for a while.
Okay. 2259. Yeah.
What are we on now? Waypoint 2? Not even.
Almost.
This is not good.
2 meters away from 1.2.
Okay.
It's not that much of a stretch.
We're gonna make tracks that way unless we see the world's most
Go to
amazing.
30 meters and that's down.
Okay, well,
100 meters difference.
we're gonna go a little bit faster now. Not
Okay.
fat. No, sorry. We are gonna work. We're gonna try and zoom less. That's my goal.
Gotcha.
Okay.
Well, the landscape's making it easy for us for a minute.
Yeah. Good job. There's a
And-
bathy pathies. So black coral.
Keep going?
Yep.
Yeah. Keep going.
Okay. Start it?
Got it. That's something in the middle, but
What? What something?
no, it was not. It was, if anything, the same yellow plexaurid.
Okay.
Most abundant
little
plexaurid you can get to a species.
Yep, deep sand.
Cody, could you bring the bright? Just it's a little too bright.
Washed out?
Yeah. Yeah.
Other bathies. Another plexaurid. Well, that's too dark.
He I think he knows that.
Wait, are you black balancing? Black balancing. Gotta make sure your black's right.
I mean, it's a thing.
It is.
Oh yeah.
Oh.
You black balance too.
So if you've recently joined us or you just want to hear us say it again, we are aboard the RV Falkor in the Central Pacific. We're diving today with ROV SuBastian and our wonderful ROV team. We are in international waters, so on a seamount that is not in any country's EEZ or exclusive economic zone. And we are traversing our way up this seamount from starting at a little over 2,200 meters and working up to a little more shallow than 2,000 meters. What do you got, Brian, about what we're doing here?
I mean, we're exploring for the most part. Like this is an area that wasn't mapped until this morning. We actually didn't know how deep this seamount was. We had a competition of guessing the seamount depth last night, and the guesses ranged from 300 meters subsurface, being the shoalest point, down to almost 1,500 meters, and it turned out to be deeper than everyone else expected, with the shoalest point on the feature being about 1,600, 1,550 meters deep. So that just gives you an idea of kind of the state of knowledge of the deep sea. Is we have satellite altimetry data that gives us a rough idea of where the seamounts are, but it can be off by the order of kilometers or miles. So we've literally kind of we knew this feature, you measure it, but being several miles, 5 to 6 miles, 7 miles, and along its longest axis, and we weren't sure how deep it was either. So we literally, more or less found a new mountain that's over 3,800 meters off the seafloor.
And we're exploring a teeny tiny itsy bitsy little sliver of it. But it's pretty awesome. Whoa.
Just some dead sponge, it looks
I
like.
know, but it just
I know, it always looks weird.
caught me off guard.
So most of what you're seeing here is probably some type of basalt that has been encrusted with what we call manganese crust. And so the underlying substrate, these were shield volcanoes formed by Pacific hotspots on the order of many tens of millions of years ago. And then over the time, you get certain different precipitants coming out of solution and welding onto the crust. You don't actually seeing the basalt here. You're seeing this crust that has grown over millennia.
Rate of precipitation is like a millimeter on a million-year scale, so it is not a thick crust, but it has taken millions of years to acquire, or for that to precipitate out of the seawater and form that crust on top of the rocks. And so some of we don't have a geologist on board with us here, but we are collecting rock samples and taking those back to geologists with the United States Geological Survey so that they can do analysis on the mineral composition of the crust as well as the underlying basalt.
Yep.
And now, actually, I was saying it was underlying basalt, but now that I look back at the map and realize where we are, so because this is a flat top feature, it was once emergent, and so it was above the surface and eroded away, and so actually beneath this could be a sedimentary limestone that was laid down by ancient coral reefs that then has been manganese encrusted. I have to admit, looking at this, the underlying texture looks more volcanic to me still. But as we move up and get onto the edge of the top, in theory, it should switch into a some kind of carbonate base that would have formed when the seamount was shallower millions of years ago.
And on watch right now,
I'm a PhD candidate at Boston University.
and ROV guys, you want to introduce yourselves?
I'm Jared Easterwood. I work for SOI.
I'm Cody. I'm co-piloting. I also work for SOI.
So they are doing all of the flying and the controlling, all of the actual hard work down here in the deep sea. They are fabulous to work with. They try and accomplish any and all of our goals, even if they're a little crazy. And then we try and be as appreciative as we can to them for all of their hard work. What is that big anemone down there?
The big purple one?
Yeah. Do you know?
Don't know it.
We collected one.
Yeah, it was named by
It's
the
good.
French.
No.
Hey, don't have that too long. I remember we
Do you want to eat dinner? I'll stay hang for a bit.
Is it dinner time?
It's 5, yeah, is it dinner?
No.
5 to 6?
5:30.
5:30.
6:30, never mind.
But yes, what's
Yeah,
dinner?
when that time comes, I thought it was 5 to 6.
It's a,
That's fine.
yeah, let's take just a zoom on that coralline star.
I'm talking about one of the big coral reefs. Yeah, okay.
We haven't seen any scleractinian corals, not even cup corals,
No, I haven't seen a single cup coral.
which is surprising to me. It's one of these big glass sponges that have, and then as they grow, they are essentially trapped inside of this sponge lattice. There are some cultures that will give this sponge as a wedding gift because it apparently represents never-ending love or something. To me, it feels like being trapped, but to each of their own. We have some more corals coming up around this ledge. So it's, we had like quite a bit of pretty bare rock surface. And then looks like we've hit a nice little biodiversity spot. There's the anthomastus down there with the polyps pulled in. So that's why it's a mushroom coral, because when all their polyps are retracted, you have that little pink ball. It looks a bit like a mushroom.
Nice purple crinoid on that.
Yeah. We have some type of primnoid. Oh, and there's another little coral with an astroschema on it too. A number of, okay, Tim,
Yeah.
would this be of interest for your duo and RNA later?
They're actually an associate on this side because it looks like there's a little crab and a crinoid in the bottom in the middle.
Oh, I didn't see that. crab?
Oh yeah, I got something long armed.
Yeah, but down here you
Yeah,
don't have
yeah.
anything.
It's just
No,
not
you're
an
right.
even half. I'm just want just take a look at it before you decide. But you're right. It fits the top and the. You know, you're right. It's all the way down at the bottom. Yeah, I take it back.
Or I mean, it's just you could
Yeah.
snip
It's
some
true.
that definitely
You could.
doesn't have an
Just matter
associate.
What
Yeah.
we wanna do, that's all.
I think we should do it
I think we should do it too.
if
Yeah.
you want one. Okay.
Yeah, do
Is
it.
this something that we
Try
can
it.
sample?
Let me see if I can get
Okay. We're gonna, if we
far off
do
the wall.
sample, it's gonna be a couple different samples from the same.
Okay.
If they have to, you know, if they have to.
This is gonna be a hard spot to sample.
Well, we're gonna let
No,
them.
no, I know. Just
From the square, if thrusters are blasting people in the water, I've never encountered that, but, yeah.
Pretty good. The earningators just so heavy.
What do you first, where was this thing off?
So where the brittle stars are, the top, we would want a piece of that with the brittle stars on it, and then from the other end, where there's none of them.
So like here at the bottom?
Yeah. So like.
This is another, this way, that way we can set up.
Yeah, so like, we would want some of this, for those stars, and then some of this, where there's none.
But if you can't.
Turn in.
Really accurately measure your distance you're traveling.
Well, yeah.
No, I mean, if we don't do a transect, it's fine.
Can I tell you one more thing before we go?
Sure, absolutely.
This is gonna
That's
go
why
in one of the
I was
two of the
hoping,
different quivers with RNA later in them.
Okay.
you know,
So if it's something where you
there's
need
always
to
a chance
pop a lid
if
before,
you put that much
Okay.
sediment on these
you would
seams,
wanna do
even
that.
if you put down on a relatively, more modest ridge,
So two Arn.
you're gonna have more opportunities
Yes.
to do
So-
transects, but
Arn-
you can't
Arn later?
transect.
Arnold.
I've never
later.
seen Arnold on paper ever.
A transect.
So we go for 23 and 24.
Okay.
I've never seen anyone publish anything about a vertical transect for quantitative purposes, other than for mosaic.
Do we not have a physical basket map? Am I?
it's taped in already.
Oh jeez. Okay.
If it's vertical, then it's a flat plane.
Yeah, the Twitter happy.
I guess no, but not forever. I mean, not for 100 meters vertically. And
Oh, here it is,
It's really
okay.
hard to control your distance from the wall because.
So, yeah, so I guess I'm thinking vertical walls in the land canyons, where I know that they are sheer for quite a way,
Yeah,
but I hear you, but I also think that we need more miles. There's always gonna be a slope unless you go to the.
So for those of you who are watching at home, what we're doing is trying to figure out if we can get the vehicle in a stable enough place to be able to work the manipulator arms. Because with two pilots, it takes more than four hands really to do this dance, because they've got to maintain flying because we can't land because we're, 19 meters above the seafloor. So you got to put a skid into the wall here and kind of balance on that one point while operating the thrusters, while panning the camera, while moving the manipulator arms, with room for the basket to come out to be able to put the sample into it. So it's an extremely complex dance here that Cody and Jared are trying to work out how to do while hovering in mid-water, to be able to do a fairly complex sampling operation here. So
You're only seeing. You're not.
So
So
we
it's
always
gonna-
defer to them. We ask them if we can do something, and they assess the situation.
Going to never get all.
At some level, don't you have to do it randomly to be a real, because you start talking about.
You got it.
That's all
Yep.
bias.
All the more reason to if you're really going to get a spatial
Like it.
variability of the communities in the Seamount to look at more transects. More club.
You know, internet UV.
Well, that's the go-to.
I'm assuming somebody.
If this is too, reach.
Love it.
We'll make it happen.
Okay.
Tim, do you want to pick out your spots?
Go ahead, Corge up. Sure.
I let them know generally what the regions
So
were.
they're
that
going
you
for
would-
the
Go on.
if you can't, yeah.
Yes, up top there are more. There are more of those ophiocanthids. Not that one. There's,
So I'm gonna
I
go
thought there
for
was
24 and 23. Is that okay, guys?
That's the ones that are that have the red marks.
That's
Yeah,
yep,
you
that's
want-
what you want.
that's
That's
what
what
we
you
want.
want.
Yes.
You
Yeah.
want the red labeled ones on these. So it's. I thought I saw like 3 if you can't.
There-
They're
they're
off
up
the screen.
top.
Up.
Show it up in there.
I lost my pen.
All right. You want to get a good picture right before I go in there?
Yes,
So
we
it's-
do. Can we pan up a little bit so we can see those? You correct me if I'm wrong,
No, you
Tim.
got it.
What about like right here? This
Can't see all the branches
Right here.
beyond that, but yeah, that was good.
or
Okay, here.
or look here.
Good. You show
Can
us
I
which
show
one?
you?
Yep, good.
Yeah, it's good. It's got multiples there, right?
Yeah,
Right
but this
there,
is
but-
pretty big. You see this? This is not going to fit in your quiver.
Well, I thought it stopped right there. Sorry. Yeah, no, that's too big. That's too big, right?
Do you want to take up
Up
here?
there. Yeah, it would have to be.
Okay.
Can we snip right here so we get this
Yeah.
buddy?
Yep.
Okay,
Works for me.
got it.
Clear to take a sample.
You get some snaps.
Get some snaps
Clear.
in?
Yep.
All
Yep.
right, going
All
forward.
right. Thank you.
You know, the way your eyes work, you have stereo vision, which helps you with depth perception. But because the pilots are working on cameras, they lose that stereo vision looking at the screen. So they have to have 2 different monitors from different angles in order to see the different axes that they're working with. Because your depth perception is lost on this kind of angle. So not only are they looking at the camera you all are looking, which we call SciCam, they've got another situational awareness camera up above that gives them the left, right, and depth axis as they're maneuvering through it. So they have to bounce back and forth between the different cameras to be able to watch where the manipulator is in regard to the coral.
Give it a little time so it's-
And now you're gonna be, look at this one, Cody? You're actually on come down just a little bit more if you can. Open up just a little bit, head on. Just pitch down.
Stick it.
Right there. Alright, and just a little bit more. You got any more arm or no? No? Okay.
Go for it. Go for it.
Alright, capture the outsides. And then swing it in. Here, do you wanna, here, let's do this. Let's do this.
That's it. Nice.
So what you're seeing in that shimmer that's coming out of the quiver there, that's a preservative that helps us preserve the RNA in it. So it's called RNA later. It's denser than water, so it sits in the bottom of the tube even though we've pulled the plug. And so we've got these specially marked quivers here with the environment.
Essentially
It's-
just very salty
Yeah.
water.
This is going to be sample 10, right?
Yeah.
You want this little piece too right here?
No, it's
That's fine.
okay.
No.
We don't need that little piece, right?
Cap it.
Nope.
Yeah.
That's great.
Okay.
Nice. The sample number. Oh, sorry. Wait.
FK010.
You're putting the FK on there?
Yeah.
FK21.
FK21, I'm sorry.
Okay.
010.
010, correct. Quiver 24.
Sweet.
Okay,
Nice.
Nice job.
well done. And then you wanna go for?
And then we want to go for 20, yeah, or just another RNA later one.
Okay, so, yep,
Whatever's easiest.
I'm good, bro. Yeah.
Nice.
Okay, and then just anywhere over here?
Let's get one without a.
Yeah, so like this kind of, these forests.
Yep,
Yeah.
Yep, like right
That'd be
there
great.
somewhere?
Yeah.
Yeah.
Just
Single piece?
same kind of thing, yeah, a couple little sprigs.
You wanna get a snapshot of it?
Yep.
Sure.
Before we grab it?
We do. All right, go for it. Would you put a list of change to
So
make
you want a couple of us?
Yes, please.
How about just right below that 4?
Yep, that's great.
Perfect. Yeah.
to
Great.
Right there?
and
So you want 4 software?
Sorry, let me write this.
There we go.
That's right, yep. That's good, that's you. Got it. Yep.
Nice.
perfect. Oh, you almost got a
Close
bonus.
the jar.
Look at that.
Come on, buddy.
Where'd that crap come from?
He was over here, not on the arm you're looking
Right.
for, but
Yeah.
was on the main.
Great
Off of
job,
arrangement, yeah.
Cody.
Always on the basket. Always. That's 11.
I think so.
I'll just jam.
One.
0110.
Whoop, baby.
And Quiver 23.
Oh, that was a grab off the brazone up there, right?
Bring the fortune.
Awesome.
Alright, happy?
Yes, very
Yeah, cool.
happy. Thank you.
Can you pan up for a second? Just
Yep.
What do you
Alexis,
want?
can you pan up for a second? Just
Oh,
to
yeah.
Capture the colony again.
Okay.
Where things are right now.
Can you remove the additional text?
I know, I
Got
know,
it,
I know.
okay. I wasn't sure if you wrote that down.
Oh, I didn't, but I just.
Good?
Great.
Okay.
Yep.
Were we doing pretty good with covering ground before?
Yeah, we're getting
Okay.
pretty good.
What was it? Remove the additional text from what?
Remove the required text
Oh,
from
okay.
sample in situ.
Okay. I would say probably next time we're somewhere that we feel is high density, high diversity, we do another
Jump on the
water.
bottom.
Yeah.
Gonna keep on trucking?
Yes,
Yep.
sir. I think someone in the chat is asking what the solution was that was in those quivers, like why it looked wavy. Is that the right description?
Yeah, the mixing.
Shimmery.
Yeah.
Primnoids that have.
See, wait,
Oh,
predatory
yeah,
sea star.
okay.
Stop here.
We'll do an image.
Thank you.
We've already started the
I'm
work.
sure you did. You know what I just did to you? I did that thing.
Oh, I found my pen. Now you can have your
Sorry.
flag.
Look at it.
Yes.
Just
Well,
look
and
at
also
it.
look at the other one. So it's not
Denuded.
just the same
Yeah,
colony.
yeah. Can you image all of it? All of the grazed areas for me?
Do you want them like very close images, or do you mean like a
I want to be able to estimate percent grazed.
So, we need
a
full,
flat.
full
Okay, we
eco
need-
gig treatment.
okay.
Sorry.
No,
Very
it's okay.
No,
exciting.
it's fine. Just-
Core livery.
What time is it? 0.
I'd much rather take it off of a primnoid too, rather than a what were we on before?
it was a hemicralium.
Hemicralium. Yep, it'll be easier. Less brittle.
Wow. Yeah.
See it everywhere.
Yeah, you're right.
I swear to you, my first time diving, I was working, or not diving, my first major project I was working on, I was in the Smithsonian in Belize. And I was with a really prominent coral scientist who said, I've never seen anything eating a coral. I was like, come with me. And after that, they were like, it's really everywhere.
Look at the tips.
It jumped from branch to branch at that certain distance away from the thing, so that all the tips are eaten,
Yeah.
but not the bases.
Well, what do you think about maybe the fact that it fell off when it got to the top and then just
But you
grabbed
see what
on
I'm
down
saying
there?
about the
I
tips
know, I totally
being
do,
wavy?
absolutely.
Because I've never seen
Yeah.
that.
No, it absolutely.
Nice.
Alright, how you like that?
I like it a lot.
You wanna see
Let's
pictures
get
real fast?
let's get some good images of this,
Yep.
and then we'll do the collection, and then we'll image the other ones.
Yeah,
Is that
perfect.
good? Okay.
Where would you like him?
In a quiver.
And a quiver.
If he'll
Will he fit in a quiver?
fit.
We
looks
could
like-
try.
Or you could put them in a bio
Yeah,
box.
we could try. I can open up one and
See if it.
we've put them in there before.
Okay.
And we put some big ones in there before. They just kind of conform to it.
Okay.
Look at
And if
that
not,
beauty.
Just open the bio box and open it.
Sounds good. I
Look
trust
at the
you.
great distribution
Ah.
of that.
Beautiful. My coralivore cup runneth over.
Yeah, it's a good.
Multiple times.
How about this one?
Okay.
So these are, this is a primnoid, so not a black coral, but what's known as a golden coral. Oh no, that's Chrysogorgia, sorry. Primnoids have the more characteristic of like these very scaly polyps. So they are not as fleshy as you see with some of the other octocorals. But we've seen corallivery pretty, probably pretty most commonly on primnoids. Is that a fair statement?
I'm honestly not sure.
Okay.
Yeah. We're gonna take some of the coral too.
Oh, okay.
Thanks.
So maybe we just go. Let's see. Is that two separate collections if we want the star and then
I would
the
su-
coral?
Yeah, I
I
would
would
suggest
go
so.
for the star first. Just so we
Yes. So
Yeah,
let's
we don't
put,
need them together.
Let's do that, but put them both in the same bio
We want
box.
them both.
Okay. So go straight to bylaws?
Yeah.
No problem.
So before we start the collection, can we zoom in at these tips that are waving?
Yeah.
You mean the left ones?
No, the
dead
ones
tips?
yeah, with the ones with the
Sorry,
angle
Nick.
that have no tissue left on them.
Those ones right in front.
Just a little left side. There we
Yeah,
go.
on the left.
Okay.
Yep.
I mean, it's like they've broken, but they're wavy. That's what's like. Even if they were broken, you would just see a flopped stick, right? But it's strange.
It's like the tissue. It's
like
like the
the
skeleton,
skeleton
has lost
has
its
also been-
structural integrity.
but there's still a
and
sheath.
some are broken, but it definitely looks like it's lost its kind of rigidity.
Yeah.
Is that
It's,
kind of like the ones to the left
You
are
know-
doing as well?
You see this here too, like they've been cut off. They've lost that.
Yeah, some
They
have been
just
cut,
finally
but some
broken
are
off,
just
right?
kind of like yeah.
Somebody wanted dinner.
it's just a strange. I've never seen
Yeah,
that.
I've never seen
before.
that either.
So
Stars just got full.
yeah.
So you can have it.
I can see this. I can see the stars breaking out.
Yeah, I
me
can see
too.
it. I can see it breaking it too. But
Yeah, but not that
but
flimsiness.
then yeah, of course it's not like it's just broken and it's a rigid piece flapping. It's like it's lost its structural integrity.
Yeah. Because it's been completely slurped by that evil Posoma.
All right, I think we're clear to start collecting. Correct?
Yeah.
Yeah.
Okay, biobox.
Bio box. One that's not used, obviously.
Wow. Look at that.
That
I
one's
know.
already denuded.
It's almost entirely. Who wants a Pronoid for dinner?
Not me.
It's cool to watch the
Torment.
all the ophiocanthins move over to the other side of the colony, right? From where the
Oh.
Posoma is. It's the first thing I noticed, right? That they're all getting away from that thing. Right?
I'm trying to figure out. Are there like? Is this one colony? Is there a colony, a separate colony, or is it just like a branch of that colony?
Little gut contents.
There's
little
a
stabilized
split here
tips,
with two, and then there's a second colony down below.
little
Yeah.
microbial swabbing.
Yeah.
All the good things.
I know.
Look, by taking this one sea star, think about how many corals we're saving.
Oh God.
It's all of who you're rooting
I'm sure
for.
this colony will be very appreciative. Thanks a lot. We should come back and see if it grows back.
Thing is, I don't think they're healing. I want to go look at the other colony just for that reason, because the coralivore not on it.
Yeah.
There's still tissue there. It's sloughed a little, but it's not entirely gone.
There we go. Yes, you are done with dinner. The buffet just closed. It's no longer Vegas. The buffet just closed.
Oh, Tim.
So I'd like to get a zoom of where that was and look at where the
Me
tissue,
too.
the dead part, and the
Yep.
live part are together.
Me three.
I wonder, they invert their stomachs, right? So I'm wondering if it's.
Yeah, if you put some coral in there with it, it'll just go back to munching. They're gonna throw it in the same bio box.
I wonder. Probably not. I think.
Yeah.
Be interesting to see.
Exactly.
What do you call that bio box?
Biobox 3.
Okay.
Which
George.
Box 3. We got all kinds of numbers in the bio boxes, by the way. H on there.
FK21
Yeah, we gotta sort this out.
012.
I think there's 3 chambers. So
It's okay.
yeah.
and then what do you eat?
This is why we do a
We
sheet
often
count.
Yes.
Yep.
to be really
Do you want
correct. you
Randy
say
and
core
Tim.
Ran-
metal.
Oh.
do you want live tissue, dead
Can
tissue,
we
both
zoom,
tissues?
Can we zoom in first and see?
We want to zoom in first and see
Sorry.
where the star was to really take a look at the
the transition of what happened.
exactly.
Look for teeth marks.
Wasn't it to the left?
Alexis, I'm sorry. Can I drive for 2 seconds?
Sure.
I want to
Well,
play with the camera. Here we go. Remember how this goes. There we go.
Shit, honestly. I'm not
All right, Tim. Here we go.
It's indicated there. Yeah. It was far left, correct?
Yeah, but see, I'm looking at this sheath that's still here, right at the edge. I know this was a back one, but it feels like, you know, so the star wasn't there at that exact moment, but it was there eventually. And then, take a look. It's the same thing here, right? You always have like a polyplus sheath, and what I'm trying to figure out is what's new growth versus what's partially eaten. See here, here's a really clear example. You have like a sheath of living tissue over the calcium skeleton, right? Or calcite skeleton that doesn't have any polyps.
So I thought it would mow down. I guess I want to see where we just pulled off to see what was most
Well, that's
recent.
right here.
That's only we could.
So no, that's right here. Right there. And it's like in process, right? So here's a partially eaten polyp.
I think where the guy just was right here, right? That's
Well,
right.
hang on. Yeah.
No?
Yeah.
So I was looking for there is a sheet there,
Yeah. Exactly.
followed by polyps.
Same thing. Let me zoom out and see if I can get it. Okay, so and so it was right here in the middle. Very
Yeah.
cool.
Okay, so there's a.
All right.
So take the polyps first, and then come back and do
Yeah.
the rest.
Yeah.
So what exactly do you want?
It doesn't matter. Just take something. Maybe take these branches up here.
But so
If
you
that's
want
easy.
grazed and ungrazed areas.
I don't need grazed and ungrazed areas. I just want for this. If you get them, I'll use them. But I just want what I'm looking for
So like those
primarily.
two right there.
If you think you can get, if you think you can get this by like pulling this right here and then taking this whole section, that would be perfect. Then done. Then it gets everything. Thanks.
And there's no colonization, anything else, there's no particles here.
Are we going to consider this a combined sample, or are we going to call them two different samples?
No, call them. It's a combined sample. They're going in the same bio box.
Okay.
No, it's two different.
It's two different samples, but in the same bio box. Sorry. Same.
But wait, so it doesn't. So that's
They'll have two different sample numbers,
two
but
sample
in the
numbers.
same bio box.
That's fine. I just want. But
Yeah.
you want this.
All right. Sorry, Alexis. Oh, I'll let you back. That's fine. Very excited.
All right. Randy, have you eaten?
What?
What was the sample? I saw the-
12.
What, 12?
Just the sea star.
Yep, and then 13 is the.
Is just the host.
Is it on the same file box?
Yes, that's fine.
Got a snack for you, my dude.
Yeah. It's got a couple more hours in there.
Open it up.
Oh, it's there.
FK2113 in the box.
Mm-hmm. Nicely done.
Good job.
In box A.
And then Randy wants to get images of these other
Yep.
skeletons.
Yep.
Okay.
Images of what? Sorry.
This coral right here.
The one right in front of us?
Yeah.
Okay.
Yeah, but we might need. Let's see we might need to reposition because we want to see the whole coral, so I can't quite get everything in view from this angle. Tilt up and just make sure we got everything we want to on that one. You can, yeah, there we go. Just hold that for a second. Let me grab that.
Okay.
Okay. Yeah, so I think we've got everything we need there. So yeah, so I believe there's 2 or 3 colonies off to the bottom left of the frame.
Okay.
And if we can try and get like planar shots of the whole thing where we can trace what is dead and what's still got life on it would be the goal.
Do you wanna? Yeah, I'll eat. Do you wanna take my spot?
Ron, you can go if you want.
Do you know what we're gonna do with the next 3 coral head
I'll tell him.
corals, Tim?
It's up to you. Whatever.
We'll tell you. So we are going.
I'll stay because we're in the middle of the operation. You go, and then. So it's dinner time here on the ship, so we're organizing watch changes so we can rotate through dinner. We don't need to hold these shots long, so if you can,
I'll just fly 'em.
if you can fly them,
Yeah.
we can probably hit that one, that first one from this angle right now.
Okay.
Stripped bare.
So we're trying to do an eco-gig style whole colony shot so we can trace the
I see.
dead stuff.
Yep.
That was just taxi driver.
Do you want me to do anything with the Zoom or just like that?
Yeah, I think that they're involved in here right now. Okay, I think we got that one. If we can pan to the one on the right and get it full frame.
Well, it's kind of hanging.
Alright, I think I got that angle. We might get a good angle if you can pirouette around to the right and then shoot
Okay.
the flat side from the other side would be great. That angle's perfect if you can just zoom out just a touch so I can get all the tips. Beautiful. That's what we need. Alright, I think we're good here.
Okay.
ROV, that's an updated waypoint for just slide back towards it.
Okay.
Thanks, Tony.
All right, y'all just want to continue now?
Yep, I think we can continue on. Thank you. That was great.
You're welcome.
as we moved away from the kind of the ridge intersection along the top.
Grazed colonies are all the same from NOAA
Yeah.
also. That's a
So that one's
pretty well hit
dead
one.
and pretty
Yeah.
well grazed as well.
Do you want to stay kind of on the edge here, or do you want to go up anymore? This looks like top of the ridge, right?
Yeah, I think we're just working around the edge, so we can stick our head up. But I suspect we're gonna turn into flat sediment real quick if we get much shallower. But I mean, if you want to.
I mean, I don't out in the distance.
Even these flat surfaces, the sediment drift's pretty thin.
That's what I was expecting. I mean, the current's
It's ripping.
pretty good here. So any sediment that falls would be scoured away, except for the smallest smallest amount. You can see ripples there too,
Yeah. Oh yeah.
indicating the wraparound current around this feature.
Yeah, so the ripples are indicative of movement, and the shape and the direction of the ripples can give you some idea of the prevailing current in an area.
There's an isodella in the
Yeah.
background. It looks like that's one we haven't seen before. Do you see one of those yet?
We saw something early in the dive that was.
One or two, the whole dive species, multiple colonies, but this is the first that I've seen. That's this isodella. Really bizarre kind of branch, the main base.
Yeah.
It's pretty tortured. No, I think that's okay. Wonder if there any associates on that. It looks like there might be one. But it does look like it was growing up in one direction, and then all of a sudden
Yeah.
prevailing current, which tells you a little bit of something about the regularity of the current here, is that it's pretty much unidirectional. There's not many indications that the current is variable here. It's pretty consistent.
Yeah, I mean, if you look, you do see some variation. It looks like scouring around. It's either right to left or left to right. Ooh, there's an umbellula. Always love those. They're super pretty. I feel like we're seeing more dead sponges than live sponges here, these big ones. Just reading the comments on the YouTube feed about the eating only certain number of polyps and then moving on. We don't know. We're just beginning to start looking at coral livery in the deep sea. It's been completely unresearched in deep water. Parasites kind of evolutionarily being optimized to take as much energy from their host as they can without killing their host. So there are documented cases of that in other environments and other parasite host pairs. We don't know if any of the deep sea corallivores actually do that or may do that. Ooh, snapped blanket eel. Or is that a snapped
Yep.
blanket? Yep.
Yep.
Yeah.
Cutthroat eel. Haven't seen many fish this time.
No, that's only the second I think I've seen. Not sure who scared more who on that one.
Right.
I mean, so now that we're on top, there's a fair amount more sediment, but you still have places that corals are
It's on hard
attaching.
grounds, absolutely.
And there's a unbranched bamboo coral right there, for example. Got another bathies in the background there. So that there are slightly different corals up on top here, despite there being patches and pockets of sediment. In fact, that you add in the sediment, you gain the sea pens, for example, the umbellula, which.
There's another Arcididae.
But you're also seeing bioturbation, right? So that's
Darkness.
another piece of the mosaic of a seamount habitat. Where you have animals that are in the sediment that are disturbing the sediment. Worms, for example, burrowing in and feeding in the sediment, or filter feeding, suspension feeding from within the sediment. You have an entire different habitat that's different from the hard substrate that the rest of the seamount's composed of. There's an anemone
Can't tell
down
what
there.
that is.
Yeah. A different bottle brush type Chrysogorgia over there too. Got so many of those.
Whip.
Heteropathes, black coral too.
Come all the way to the top of the thing. That's another.
You want to keep going up?
Is a stalked crinoid coming up?
That's a. Yeah, that's a
it's
stalked
crinoid. I think that's the first stalked crinoid we've seen this dive. You did one? Okay.
You want to keep going up?
Go eat dinner, guys.
I think, I don't know, I'll let Randy make decisions.
Well, you want me to do what?
We're just trying to figure out, so we've been coming up, we've kind of lost the wall, and we've gotten up a little more on top in the more heavy sediment. We're just discussing whether to continue up
There's
or
still, there's still corals
There's still
here.
corals, absolutely.
Yeah.
When was the last time you fired a water sample?
We,
At
a while,
the-
at the hut.
Oh, God, I love those crinoids.
Right, I'll leave it. You can figure that out.
Leave your computer up. Let's just keep going up. But,
Fine,
we can. Okay.
Like tomato, crinoid.
You know, I just want to point out the fact that I've never ever thought about how great of a question it is to ask, are crinoids like tomatoes? And if you're going to say, are crinoids like tomatoes, then I think it's completely apt. And they're red. All right, so where are you? I don't know where you are.
Northish, I mean,
Okay.
going uphill. Do you want me to make my way back towards the waypoint or just keep going?
Keep going. Towards the waypoint, right? Yeah.
Yeah, well, we kind of deviated
Oh, I see, I
off
see.
the track a little bit. We're off the map up there.
You're off the map. Crinoid.
So, yeah,
Yeah, just keep
I-
going.
yeah. Can we come back towards the line in a gentle arc? Because I feel like we haven't really reached the crest of this ridge
I
yet.
agree.
It goes up off that distance, but if we want to do our dive track, we need to come back a little bit.
You know what? Why don't you just follow? You can follow this ridge up too and meet the point up there. It's up to you.
Well, that'll take this.
Oh, I
Following this
see.
will take us northwest, and our dive track is northeast.
just north.
Yeah, north.
Why don't we? Yeah, let's just kind of get there and then keep
Do
going.
north.
Because we want to see the top of this feature. It's gonna be cool. I can just feel it. Look at those tracks.
And they're just ripples, like
Oh,
from
I
the
know.
current. Yeah.
Oh, they just look cool. That's the bone from the whale fall. No, I'm just kidding. Just a dead sponge. The look on your face, though, is of like, Randy. So if you're just tuning in, we are on an unnamed seamount that was unmapped. Any nation's waters. It's ABNJ, areas beyond national jurisdiction. This is the very first inaugural dive of our expedition. We have Steve Askovich. We have
Hello.
me, Randy Rogen. We have our fabulous ROV pilots. Woohoo! Our marine techs, a whole bunch of people. 39 people on this ship who are all enabling this dive to happen. So thank you very much to the Schmidt Ocean Institute and the crew of the RV Falkor for helping us to dive this incredible piece of seafloor. So we are traversing perhaps some terrain that is a little less corally. Ooh, although
There are
There's some
corals there.
stuff.
They're just
Yeah.
different. There's a right there. That's on the left-hand side
Yeah, what
about
is it?
9 o'clock. There's an umbellapathes,
Yep.
black coral.
Towards our waypoints so that we can make sure that we hit the top of this feature. Let me log it. Okay.
So that's fine. We don't.
Oh no, I can't use a
I don't
map.
know if we need to
Here it is.
do a full zoom on it, but
Thank you.
looks good. But yeah, we were commenting. Oh, there's a
And look, there's a
larger paragorgia.
what is that? It's a sea pen
Sea
behind,
pen,
right?
yep. So at least a couple different species of sea pens so far. It shows how you get these large branching colony. Oh, there's a gall on that one too. That's a hemicorallium.
Yeah,
Sorry.
I got it.
There's a
What kind of oak is that?
Asteroschema, I think.
We don't have a hemicorallium button?
We don't. It was on the board. Didn't get translated to Coralliidae there,
All right.
but we can add it in later.
Can you make a note that we need that button? Corallidae. Oh, here it is, yeah. Hemicorallium.
But there's still an appreciable amount of stuff here, and it's different than the rim of the ridge we were just on.
Absolutely.
Yeah, it's old. Looks like it's possibly a dead sponge.
I think it's dead.
Yeah.
There's still an awful lot of it.
Getting pretty flat up here.
You know what we haven't seen any of? Spongivery.
That is more rare.
Oh, there's another stalked crinoid.
If we want to acquire the ledge, we might have to go.
What's that? Oh, we might have to go where?
That's another Bathybathes.
Okay.
We might have to go back towards the waypoint track.
Yeah,
Yeah.
let's just do it.
Yeah, that's okay. Bring it back towards the.
I can't keep track of all these. There's a lot of stuff here.
I mean, it is interesting. You know, it shows the premium of the hard bottom for suspension and filter feeding things.
Absolutely. Is that Stichopathes? Or is that just a...
We've been seeing some unbranched bamboo corals.
Yeah.
Yeah, we just changed your way.
There are some heteropathes, black corals as well. That sometimes look unbranched, like
Yeah.
I think this one. We saw them.
That looks like another, like baby Iridogorgia or
yeah,
something.
yeah,
Doesn't it?
yeah, very young.
Is there anything you wanna have a look at while we're around here? We have to get the vessel to move a little bit east before we start going that way, so.
That's all right. Can we take a look at this thing right here? This long thin stalk. That looks odd. So, what is that?
What is that?
I think it's a sea cucumber.
There's a Bathypathes behind it. I'll just log that. Ooh,
Sea cucumber.
that's another Antipatherium.
Oh yeah, that one.
Definitely. Don't you think?
Yeah, yeah. No, it's it's the same one we've been seeing for a while. This one just is unbranched.
Yeah.
And yeah, with these they kind of branch at very odd intervals. All right, that looks good enough.
You don't wanna zoom in tight?
While we have a second, we should take a closer look.
We can take a closer look at this and
Anything else we want to look at here?
do we care what kind of star that is?
That's a Brisingid, almost certainly.
Think?
I don't know.
Okay.
Can't do it much better than that at the moment, but the way it's perched on that rock screams Brisingid.
All right. Patchy. Those mucus nets are everywhere. It almost looks like it's either folded or it looks like one half of it's been sheared somehow.
Yeah, I mean, it might just be the perspective that we're shown on this side.
Yeah, it looks more folded the more I'm trying to focus it here. Yeah, it's just folded.
Yeah.
There we go. You know, I don't see them actually capture food very often, do you?
Rarely, yeah.
I mean, it's pretty interesting. I'm watching some of these particles actually blow into the tentacles and not stick.
Yeah, I mean, you might not see that upshoring, or if it's... I'm sure there has to be some sort of selectivity.
This might just not be coming from the right direction. It's coming from the side rather than down too. Absolutely.
Right. Looks good. I don't see any other associates, so I wouldn't put it up for a candidate for a collection since we've
No.
seen a few others. All set? All set?
Yeah. Again,
One
just
second.
yeah. I'll say.
So it's Parentopathes?
Like? No, it's more similar to an unbranched version of the heteropathes we've been seeing.
Oh. All right.
Have you eaten dinner, Steve?
I didn't see any crabs in there.
No, I didn't see any either.
Pretty neat though.
But we've seen a number of
Is
these.
there a smaller one there too?
No, I think it's a dead sponge.
This is
Rockworthy?
interesting. I think they're a little smaller than a candidate, but it is interesting to note that they do have a lot of what look like botryoidal crusts on them,
I'm
not almost
just saying
nodule like.
Much more pitted though.
Yeah.
This is a different.
Whoa. Hello, crinoids.
And they have a swimming law. Are we? Where are we on the map?
They can. They can swim.
We're off.
Oh, way off.
We're way off.
Oh, so we're up on the cap.
The ship's on our waypoint.
Yeah, we're
Because we went straight.
getting back there.
That's a perfectly round rock. We can maybe angle down but move.
Okay, let me just
Is
check.
that okay with you?
Yeah. Well, you still have that push core, right?
Yep. No, it
The scoop broke.
the scoop broke. We do have the push core. Actually, do you want a push core?
No, I don't think. I mean, you can try it, but I don't know if it's gonna work. But there's no other better place to try it than here.
Can we try a push core?
I'm very curious how it'll go, if it's cohesive
What
or
about
not.
how deep it?
Let's see. It's a good idea.
I don't.
Good
I
thoughts.
mean,
Good thought, Steve.
I don't think. I'm hoping we won't see this much sediment elsewhere, but yeah, that we're here,
Might as
Aren't
well.
we seeing pieces of rock underneath here?
might yeah, it's
It's a
pretty.
veneer of sediment, right?
Can you-
Cool.
Yeah, this is what this is. This is why it's so hard to interpret the backscatter and the. That's
Yep.
All right,
All right.
not gonna push core with that.
Nope. Okay. Unless you want to scoop with the push core.
Nope.
Nope.
But I mean, there are still
Yeah.
things Colin has
Absolutely.
Small stones, rocks.
Yeah. And there's tons of stuff in the sediment too. It's just meiofauna.
Thank you.
Could spin what you did today. It's like, hey honey, how was your day? You know, I poked some sediment, bottom of the ocean.
Scientific poke.
Scientific poke. Why'd you do it? To see if there was rock underneath. Sounds like another day in the deep sea.
It's an experiment.
All good. What is that? Is that a sponge?
Yeah.
Yeah, I think it is. Another bath of these. Here we go. This is the computer. Sorry, your spot back.
You could stay there. Just up right here.
I'll stay here just for 5 minutes. I'll stay until we get on something good, and then I'll leave. Again.
Sit here and backseat drive. It's more fun sometimes. Get the pack on instead of be responsible.
But the round rock pinch up doesn't get much rounder than that rock right here. On the left of the screen.
Yeah. No, that's pretty good. Round rock.
I know. Brian, what do you think?
What?
Round rock. Behold. Shall we collect one?
We should. Yeah. Yes.
This will be our second rock.
Yeah.
All right, let's get a round
Let's
rock.
Get could probably not the biggest, but one of the bigger ones.
So you think you can get that perfectly round rock?
I didn't see the earlier rock. Is this going to be easy enough to tell it apart in the same rock box?
Yeah. This
Oh,
one's gonna be
yeah.
rounder. Rock number 2.
Yeah, that'll.
What sample number is this?
You know, I have in my notes. This will be 14,
Okay.
but nobody called that basket diagram.
Ooh, I don't know. That's gonna be hard to pick up.
Feel free.
It would appear that Alexis did not.
Well, I guess it's 14. Can you go back down?
Mm-hmm.
And you can see
Maybe.
what
I
the
can't figure out
last
how to use this computer.
sampling number was.
Nope.
Although it doesn't show you that far back.
Nope.
Okay. Well, we're gonna
13 was the second from no, so this will be some kind of a
we're gonna call it 14.
Oh, that rock's bigger than I thought it was. We'll go for a smaller rock than that.
2 hands, shovel.
No, just go for a smaller rock.
You want a smaller one?
Yeah, that rock is bigger than we need.
What? Okay, you can go.
Yeah, it's.
If we get a small one, you say it's
I mean,
too
that one's
small.
just so round.
What
It's
about-
just so
What
perfect.
about this round?
Too
tiny-
small.
That's too small? What?
Yeah.
We can bring the forks over and try to scoop it on too.
No,
Nah.
This one's about
Small.
10.
Too
we're,
small.
It is on the small side. We're looking for the 15 to 20 range.
Can we
I mean-
can we find the Goldilocks rock, the perfect intermediate?
I mean, I guess my recommendation
What
would
about-
be the biggest rock you can pick up.
What about this one? That's like roundish but ovalish, so it looks like it's pick upable.
Are you sure you don't want the basketball? Because I think
If you think you can get the basketball, get the basketball.
It's gonna be fun to lug around at midnight.
We're gonna roll a rock.
There are.
I just want to point this out.
There are multitudes of choices if it
I
turns
know.
out to not be.
What about the one?
To get some better imagery, like they want tight zooms on the rock also before sampling, but we'll get it.
I feel like this is asking a lot.
we'll lift it up and I'll bring the torch out.
Stand over the top of you, torch out from there.
There you go.
Look at that.
Jump shot.
net.
That was amazing. Well done. I am super impressed.
Challenge accepted.
Challenge accepted. Which rock box is that in? It is the only rock box. We only have one rock box. Here it is. I'm sorry. I'm tired. But here we go. Rock box, rock box. Got it. And this is rocks. Check. Hey, look at that. We've checked both of our rocks. All right. Let's keep going. What's that? One of those anemones? No, it's an anemone, I think. Hanging out on the crinoid on
Looks
top
like
of an old stalk.
some sort of old brand.
Yeah, exactly. Sea cucumber. I got it.
Yeah, but these definitely look like almost like nodules.
They look so rubbly, like river rocks.
This is what I imagine a nodule field would look like.
Except there's still so much life here.
Yeah, I'm surprised. You know, there's still some. There's a
Yeah,
branchy
yeah.
thing right there.
Oh, there really is. I wasn't trying to be sarcastic.
Oh.
There's a lot. I'm never sarcastic. There you go. Another stock crinoid. Something. Sponge. It's dead. So we are just transiting over to the side to catch up to our waypoint. And as we do that, we're getting treated to this view that we weren't quite expecting, but it's really interesting rubble fields mixed in with sediment, and there's still all kinds of stuff growing on there. There's another sponge and got all kinds of stalked creatures. Glass sponge.
Yeah, Walteria type.
I was just thinking that. It's a crinoid.
Yeah, there's plenty here. It's just smaller, and I
Yeah.
wonder if it's related to the differences in flow conditions on the vertical surfaces versus
Sea
on
cucumber.
these more moderate slopes. And there's a big sponge there.
Yeah. And a crinoid. And a brazingid. Gorgeous. And there's an oak on it.
I mean, what's strange is that we're used to seeing those when we were in the Phoenix area. So far, we've only seen them on these moderate slopes.
What's that? Big fuzzy thing in the middle? See that?
Hydroid clump.
Okay.
Good.
Yep, thank you. There we go. Cute little sponge.
Looks like reflection on the case that Colin showed on camera.
Cucumber. Whoa, hang on a second. What's that?
Bridge, ROV.
Oh yeah,
Acorn?
that's right.
What is it?
Acorn worms.
Oh, it's
How many
huge.
chordates? Yeah. It's about,
Oh, yeah.
It's about. So this one's going somewhere.
Not going anywhere, no trash.
It's big.
It's good.
Oh yeah.
It's just got little sporadic.
It was totally not set up.
Yeah. It's making tracks. He's pointing us. He's saying, "Go this way." He looks like an arrow.
Or question mark.
What?
Or question mark now.
Question mark. That's true.
Or a heart. His poop and his
Aw,
tail
his
makes a heart.
half heart, half poop.
You complete me.
Aw. Okay. Well, Brian, I think I have still. All right, we're gonna let Alexis and Brian take over.
Wait, let's have a conversation.
Here we are, the edge of the world.
Have we transected yet today?
We haven't photo mosaicked either.
I haven't seen a candidate. I think this being as wally as it was, wasn't good.
Yeah, Chrysler Gorgon.
Wasn't great for either.
Do the top.
I've seen something like that. Something like that in the guide, but I was never sure if it was just a very odd branch, and now it makes me think that it's actually a branch in morphology, but I don't have anything like that.
Would you like
So
a
does that mean
sample?
You would like a sample?
Is that a long form? We would like to sample this. Okay.
Okay. A small piece.
Okay.
You don't need like half the thing, just
You got that?
a few centimeters.
I got it.
Cool.
Yeah.
Do you mean, have you seen it with these weird little side branches before?
You mean like the branch that's
The long.
half the way down?
Yeah.
That's what I'm talking about. Like,
Okay.
there's something that looks like that. It's unbranched, but I've never seen a branched one before.
Yeah.
And I'm wondering if it's similar to the one we sampled at the beginning of the dive, or if it's something really different that just is very sparsely branching through its life.
Yeah.
So, I mean, if there was an associate in there, you know, I don't need
I
the.
don't see one.
I don't see one either.
I don't need the, whichever is easiest or more convenient to.
I've been looking for
clip.
my little jelly,
So just like here, Steve.
Yeah, exactly.
because it likes bicycle here.
Or either or.
Yeah,
I'd like
yeah, yeah.
the terminal end.
What's that mean?
Yeah,
This?
that one.
Okay.
Dude,
Catch
the
up
current
eye.
is really dropped off here.
The what?
Yeah.
The current.
It's just sitting there.
Nothing's moving very quickly here, which is probably why. What's that?
But coral.
Coral eating?
Yeah, because if this was a shallow water coral, you'd be watching it come. I don't see something sticking.
So what do you say?
Well, I mean, the explored polyps are insanely small.
I know, but you'd still think that they would like
Oh, you don't have a table?
nematocysts and
Yeah,
mucus,
something.
like lots of stuff would stick to it. They would clear it if they don't want it. Right, food is so sparse, you'd think that the adaptation would be to grab it while you can get it and then discard it if you don't want it.
It's also 2 degrees, you know.
All the reason to grab it, grab it, something's grabbing it and grab it.
Yeah, but I mean, you can grab something and hold on to it. It's gonna be of good quality either way, you think. I wonder if you can be picky.
It just bugs me that I've never seen any behavior. I know their
Acts
metabolism is very
much-
fast.
Let me see what I called this last time. I have a picture of it. Must look exactly like this one.
What quiver did he open?
What?
Did you see what quiver he opened?
I believe it was 6.
Okay.
But we should double
I
check
will,
that.
yeah.
The cover to your microphone.
This here.
Just cover that.
That was it.
You want to have a little zoom?
Zoom, zoom. Yeah, let's do some.
Species 11.
Such a creative name.
Did you get some shots? Okay, let's do a little zoom, zoom.
It's either that or something like this
That looks
that
better.
branches. But this is
Good.
Pretty quick.
this doesn't.
Okay.
This has never been
Steve, do you
recorded.
need any more images than just?
Now that-
Okay.
Yeah, just close in.
Yeah.
Um,
So I'd say like here.
yeah, but this doesn't branch at all. This has never been reported to branch like that. So. Yeah, sounds good. And then the one we sampled earlier.
Yep, that looks great.
Something more like this. This, I think.
Yeah, yeah.
And it means Europe take this same crabs.
Yeah.
The thing is, this you can see from the lower gorge here, it's the same depth range as kind of what we're in. So where we're in. This one. Yeah.
Are you in control of that? On when?
Actually, it's 4. Sorry.
I just finished. Species number 11?
That would be nice. It's either that or 19. know. I think 11 and 19 have a nice ring to it, though.
What's Latin for 11?
For 11?
Yeah.
Latin?
Yeah. Chrysogorgia, whatever 11 is.
Alright, great.
I don't, not certain, but maybe. Bowling ball.
Yeah, right? They did a great job with that.
Everyone's gonna take turns
No,
trying
wait.
to carry it across the deck. They're gonna need
No, wait, the
tons
rock,
of hands.
guys, just to confirm, the rock you collected, you entered as sample 14 rock. So this would be sample 15.
Yeah.
Okay.
Oh, right, we tried to take sediment. That's what it was. I did 15 as the first board in my head.
My last request, if you see any more plexaurids, just grab one.
The yellow ones?
Any of them.
Okay.
Because they're very rare here. It would be nice to get one. Because I checked the deepest depth rating for the plexaurid that showed up before, and it was like 1,700 meters. So this would be a much deeper observation. It's very similar to the plexaurid species too.
Yeah.
Yeah.
Lots of space.
Yeah, I like this configuration.
I like the rock boxes. I keep the rock boxes.
Given that we have so many of a rock agenda, we should keep a rock box. I don't like these. Yeah.
There might be a circumstance, for example, if we know we're going to be diving very shallow, where we're only going to see carbon.
There we go.
Probably switch swap
I
that
know,
out.
we know we go higher
Got it.
samples.
Yeah.
Yeah.
Well, you know, it's a pretty big rock box. Your body amounts.
Nice.
Thank you, gentlemen.
We put half quivers and half rocks. I don't know if that's possible.
You're gonna have to wait for $4. We'll just leave it for now.
Yeah, I'm also trying to take it now.
Yeah.
Okay,
So what do you want to see from the rest of the dive?
it's a really low waypoint, so I feel like we should just do it and then,
Going over, keep going along or just?
well, let's see what it looks like. If it's amazing, then yeah, if it's not, we have plenty to do. But I don't know.
I don't know if it's going to be amazing. It could be, but I have a feeling it.
So we're just gonna traverse along this wall.
There's another one.
Kind
I'm
of
gonna
the-
snap zoom on this.
Okay.
Look, it's gonna be the same species as the other coral you just collected.
So
But it's
Can I
good to
just
know
quickly
that-
zoom on this coral? Thank you.
It's good to know that it
Okay.
looks like it branches also.
it's cool to see that Walteria sponge.
It's a little, I mean,
It's funky.
Hey, did you see the note from Jeremy Horowitz up there? Thanks,
From-
Jeremy. Thinks it's Heteropathes intricata or an undescribed sister species to this one. Might be worth grabbing it.
Which one was that?
They're very
That
few
was one
species.
before.
The black
Yeah,
coral
exactly.
we were looking at. That was the black
If we see
coral
another one, Jeremy,
a while ago.
we'll check it out.
We saw
Thank
one
you.
at the beginning of the dive. We saw a few in the middle.
not
and we just saw one
You're not
recently
grabbing
that
this.
Just wanted to
Thank
look
you.
at it. Thank you.
branched.
The Pentapathes one.
Yeah,
Right.
yeah. But it's. I've seen a number of those. They all are, as far as I'm concerned, the same species.
same kind of guy. Keep cruising. Am I zoomed in? No. Okay.
Well, the thing is with.
Don't want to be.
You want to sit up?
No, you sit. I've been sitting all day.
You have a long day.
What?
You've had a long day. Well, that's
One
pretty
way
cool.
longer.
This is right at the cap, right?
Yeah, yeah.
Nice to look up.
Possibly that could be a
We
book.
were traversing the top.
I watched the whole thing.
Me.
Yeah, I saw it.
Where did sea pandas come from?
Bamboo.
We were time ago. place far, far away called Australia.
Ah.
Upper Seamount.
Upper Seamount. Where was it? Upper Seamount? Okay, very long ago. Place called Upper Seamount.
We kept finding a whole bunch of bamboo coral that had no coral, they were just heaped up. Yeah.
Oh.
Bumps on all sides?
Yeah, it was just all gone. Like they kept wondering what's eating all the bamboo coral out. And then finally we saw a sea panda. One
Ah.
just it was really a star for the seaside, but because pandas eat bamboo.
Gotcha.
It was a sea panda.
Can we make this a little less bright? Sorry, I just want to focus on the coral.
Just to be clear, sea pandas are coralivores.
What is?
Sea pandas are coralivores.
Is this your little Webby Primnoan, Steve? Is he still in here?
Is that a promote?
Or is it bamboo?
It's bamboo.
It is bamboo, okay.
That's bamboo.
That's what I thought.
It is the polyps
Right
look
there.
like bamboo.
They're very narrow.
See him?
Yes,
see one
I
or
got
two
him,
of
I got
those.
him, I got him.
Okay. Cool. We can move on.
I agree, it's cool. I will move on.
Thank you, sir.
There's a shrimp down there, that's also cool.
Shrimp.
Do you want to see a
Everybody
shrimp?
Where's shrimp?
It's hard out there for a shrimp.
Another shrimp.
Shrimp and crinoid.
See that?
I wouldn't try to eat
Pretty
both.
cool terrain.
It is cool. It's cavey.
What do you got, Tim?
Shrimp.
Pneumatic sinus.
Ah.
You heard me say it?
Yeah, I told you.
Just said the rostrum looked a little short.
Crinoid.
Probably not in
Yellow.
nice wall here.
Little nubbin.
It's nice to ground truth and
Oh.
multi-beam.
That's
Whoa.
that's
Oh,
sponge.
it's
All up in my face. Right in my face.
Bella.
Right there.
It's a long sponge.
Trying to search right up right now.
That's cool.
cool.
Yeah, I don't know. It's the first lab processing. So, doing that at 3 in the morning was about
Yeah, yeah,
the best
yeah. I think we should
just
get to the waypoint.
Another sponge.
The respirometry experiments are not, I don't know what they're gonna look like. I mean, these things, if these ones metabolize like tropical corals, it's gonna be good. If they don't, it's
Oh
gonna be
yeah, there's a coral.
bad.
Yeah.
I mean,
Actually,
not
can we
bad.
take a look
I mean,
at
it's a-
that?
Yeah, I'm just backing up a little bit.
Sounds
Not a
good.
decent
I was
haul.
getting wrapped up by rocks.
Yeah, we do.
Oh no,
You know,
Everyone's got some.
What's in there?
That's
What is it?
Zoom. Zoom looks like.
Is that okay?
Totally
Okay.
okay.
That's a new sponge too.
It looks like somebody's living on it.
It looks like an anemone.
Ebony.
Still kind of one of
Twice
those.
so is that. Look
Yeah.
at that.
That is beautiful. Got that like honeycomb.
Never put it over me.
Okay. Cool.
What time is it?
7.
Carry on.
Carry on.
GOT.
Gotta sing the song.
5:58.
We got some bamboos.
Scram.
Shrimp.
Scrape this swimming along. Doing his thing.
There used to be a Plesiopenaeus. Then it was a Plesionga.
Shrimp. Come on, stay in the frame. Scream down. Yeah. Where you go, buddy? Where were your friends? Right there.
Any good comments on the chat?
I was just looking. We've got some suggestions to look some species which might be undescribed. We have some of the Antipotherians.
So we got friends coming in, or is it just people that want to like?
Jeremy Horowitz. Well. Well.
So come out of it.
I'm around the corner.
We're not.
Come around the corner.
You're true. You're true.
Didn't happen on that one.
You promised him.
I had problems.
Yeah, it looks like that kind of lower. Where were we when we were in like the hot spot? Like right around 2,000 meters?
Yes.
Yeah.
So, do you feel like you're looking at terraces here?
Oh, maybe. Yeah.
Right?
Yeah.
I would. Certainly, there's been some mass wasting here, but I think it's all the rocks you saw down below. But this could be terracing, right?
Yeah. But it's really thin. Really narrow terracing.
Yeah. Look at that shot, right?
I mean, this is so vertical and such a sharp relief. I feel like this is all mass wasting.
What's
Yeah.
that
Now,
mean?
but I think, yeah, it wasted now. Now you're seeing the terracing.
Yeah.
You can see the layers of it.
Oh, you're seeing the
Yeah.
layers.
The stands,
Got it.
different stands.
Got it. Got it.
Right. Ratchet.
Well, we're out of the current. We seem to have gotten out of the current. In here seems to. The current flow is way reduced from where we were seeing earlier when we saw the higher density.
Interesting.
It looks like see a sediment line in the rock down there, see it?
Yeah.
Well, in some ways we've come around the corner.
Yep.
We're no longer in that little hollow. Whatever you call it.
So, we'll hit our
I
last
found
wave.
a coral.
There is a coral. It's another chrysogorgid like we just saw.
Is it?
I don't see an associate. No crab.
Funky.
It's cool.
Another long one back there.
There's another one behind it.
Yeah.
See it?
More like the bottle brushy one.
There's an associate.
There is
now,
an
no?
associate, yeah.
Sorry.
And which one?
We're like saying
In the
exactly
right
the same thing.
You wanna go
At
to the right one or
exactly
the
the
left
same
one?
time.
Go to the right one, please. Ah.
A little bit more zoom in.
Yes. Very small. Europtychus.
Is it your time?
We're asking you.
Yeah. I'm up for anything. We're going along the same depth
Blocking all my
now.
light.
Yeah. That's okay. We're pretty good.
It's like a really big rock.
There's no more vertical relief to cover now it's here. I don't think.
We're good.
Like that.
I would say let's run it for a few more minutes, but if it's more of the same, then I think we're just going to run in.
Quite the overhang right there.
It's all stolen.
I
I feel like it's always.
Wait, is that was this the stolen first stuff that
I'm gonna. Okay.
a little bit more?
That is. Does look like
Look at all those
Good
little.
call.
little
Steve,
stoloniferans.
you see that?
Yeah, I see. Good.
That's why he went from rat to see appeared, I'm sure, at the moment.
Did he?
It's frozen?
No, it's okay.
Just look at that though.
It really did.
Steve, do you want to try and get any of these?
Just scrape them.
Here. Well,
They don't have suction, so we can't do
oh,
that.
that's right. We don't. That's
yeah.
right. Section is offline. That's right. Never mind.
Okay.
You can push for anything these days.
I don't know that one. I've never seen something like that. There's no good.
No, yeah.
Those are very little tiny stone
They're
nuggets.
all very fun.
Yeah. That'd be literally.
I know.
Yeah, they're reporting all three of them.
Central.
Exploring.
All three what?
Sponge.
PPRO views, the 4K direct, then the 4K
Yes.
CHD.
A couple little chrysogorgids down there.
Six.
Yeah, 4K downgraded. Which one do you want to look at?
This little guy right here first.
Ooh, who's this?
It looks like if I hit that rock, it will fall down.
They rocks on.
You break it, you buy it.
I break this thing all the time.
I meant the rock, not the vehicle.
You break it, you fix it.
That's it.
That's how that works.
Tim.
Yeah.
Or if we go to midnight, the other ship fixes it.
I know. I understood the midnight.
Second I took the picture.
Yeah.
No, I
well,
don't
that
see
broke it,
any.
Sorry.
What did you do?
But that's a nice
Don't point it at me again.
Your
coral.
See, don't take it more at it.
I did.
No, you're good.
Do some more.
It's like wedged in there.
Yeah. Oh, here's an interesting. Can I?
You wanna look at that guy?
Yes, please.
Keep it as the launch one. Okay, we'll take the launch one.
Well, I think they just pulled it up on the wire cam.
Two crabs.
Yeah, I think so. That's kind of why I want to look at it.
I think so. Looks like there's somebody home on this one.
There's two buddies home.
Two crabs.
Roommates.
These Euro tigers.
I would think so, but I can't really tell.
Oh, most assuredly they are.
Okay.
I don't know how we can see them.
There you go, Tim.
Red face.
Red face.
Red face more. Alright.
Is that the first time I've seen two?
Yeah.
Is this
There was one
Vitellogorgia?
other one.
Because I feel like that
It's
very
got
honeycomb.
It does have the honeycomb shape to it.
All I'm gonna do is go look through.
That's literally all I'm gonna do.
I got long arms.
Is that all you got?
You want me to go more?
Yeah.
Okay.
I want to see his eyebrows.
There you go.
Yeah, hold it there and I'll try to
Yeah.
Look at her feeding. That's nice.
Beauty shot.
Already clicked.
Looking for spines on the back. See any spines on the back?
Of what? Of
The crab?
no, I do not. But I can't really see the back of the crab.
Alright, I got it.
for a second.
You're good. Yep.
Make him get a picture from himself.
You're good. Has that branch on the lower end of the stalk too.
Yeah.
OMGs. Okay.
This one has
I don't think-
we got a 10
T-minus 12 minutes.
So you don't like these guys anymore.
I mean, we
right?
do like them, but we just don't need to zoom on every single one.
You don't like them anymore?
Oh, it's.
Oh,
Ooh, an
Oh,
Iridogorgia. Good job.
Is that Metallogorgia?
Let's say that many
No, that's a
No, it's an Iridogorgia.
I remember, you always got it wrong because I thought it looked like a symbol and it reminded me of.
Oh, yeah, that's right.
Oh.
You wanna look at the anemone real quick, or you wanna go?
No,
the
let's
ri-
go right to the coral.
Go to the core.
so biased.
The anemone's cool, but.
Coming in slow, Cody, coming in slow. I'm gonna
Spring
rotate.
grab.
I'm gonna try to get a little bit more of that black background out. I'm gonna rotate to my right.
Can you iris down and crush the blacks in the background?
Crush the blacks.
Screen grab.
Is this
Screen grab.
is this
There it is.
Iridogorgia? Is
There
it wavy or is it helical?
I got you off the score.
You tell me?
I think it's
I got you.
wavy.
I'm gonna make it dusty and then you're not gonna be able to see me.
Well, you'll be able to tell a lot better if you look down the
I know.
axis.
Like the dust.
Long back. Down the long axis.
Yeah, do a nice little.
I think it's,
There you go.
I'm gonna call it, oh, you want some ESPN?
Uh huh. I do.
You want right from the middle.
No.
Yeah, we want to look down the middle of the barrel.
Spiral.
Yeah, spiral.
it does, so which one is that? Is this, Bella?
Iridogorgia.
That's gorgeous.
Iridogorgia Bella?
Still, no, I think it's Magnus Borealis.
You do?
Magnus borealis.
The coils aren't.
Can you take a beauty shot? Oh, you just did. I didn't know. Sorry.
You could take a.
Somewhere in
You
the
could.
galaxy
This would
far,
be
far
one
away.
thing. I feel like this is something for if Eric's looking for footage.
Yeah, you know what? Let me go get Eric before you get off of this.
Oh, are you sure? Okay.
Is that what you want me to do?
It's still lovely.
I just wanted to freeze it.
Yeah.
Let's see if Paul's closed.
They won't.
That I can make them close. You want that?
Vegas,
No.
7-Eleven.
Watch one of these polyps close.
They do. That was cool though. Like it felt like you're going into it.
I used it.
Set up hashtag no filter, hashtag no zoom.
Good job. What'd you say?
Set up hashtag no filter, hashtag no zoom.
Yeah.
Any other kind of shots y'all wanted this guy?
No, I think.
I don't, was Randy trying to get Eric in
No,
here?
I think we're good.
Okay.
I wouldn't
Really?
do that. I think we're good.
Alright, so we're coming off bottom, is what you're saying?
Yes, sir.
Yep. And then we just want to, once we,
I can't see the bottom.
All clear.
My altimeter can't see the bottom right now.
Oh, really?
You have to up.
Well, because yes, I get yes. Okay.
All clear.
Okay.
Everywhere.
So we're gonna come up,
I'm gonna-
fire the Niskin, and
I'm
then
just gonna go get in position and find the vessel.
great.
missed all y'all.
You have 3 y'alls in the same sentence.
Hey y'all, hey. Hey y'all, hey.
Hey girl, hey.
Y'all listen, y'all rascal. Coming up now. On your purpose, we know this.
What's our total sample count?
This will
16?
be 60. The final water bottle will be 16.
You normally
Yeah.
It's good.
Yeah.
It's really good. There's a shot at the top. Look at that.
Yeah, it is really just a.
It's super sheer. It's all sheer all the way around. Look at that.
It is so dark.
It's almost like it's the deep sea.
Set, what do we call it? Our lines,
Completed our
course.
track line.
our course. And so the ROV pilots are positioning the ROV in the right position for the vehicle and then, or for the vessel, and then we will take one more Niskin sample, so one more water sample before we come up, but then we'll head up through blue water for a little while before we hit the surface.
Okay. I'm taking 20 unless you need me. Shrimp.
Shrimp.
I think we should add a shrimp to.
There's not a shrimp.
We
And
just put it on. We just put it under
Yeti
crustaceans.
shrimp?
Yeti
Oh, wait.
shrimp?
Always out of focus.
You got one left.
Alright.
Let's start coming on.
Look at that linear feature. How straight it is.
Yeah. That is like those lines.
Look at those lines.
That's what we were just saying.
Look at those lines.
You wanna look
Guys,
No, really,
No,
look
we
at those
don't.
lines.
Really,
No,
look
really.
at-
Good grab.
Sorry.
A lonely spot.
Last one.
Blast off. Great. What was that? 16, 18, 06, 18.
Bring you up to 1900, I guess. Maybe that would be 30 meters. off
Yeah,
the
that
top.
sounds good.
What's our altitude now? 10.
No, well,
Basically any time.
30 meters off the bottom. Right?
you want 30? What do you want?
You
How?
can take a Niskin now.
You
That's
can
fine.
take a Niskin now.
Okay. Awesome.
We want the now Niskin. There you go.
I want my Niskin just
Make a note, 10 meters off the bottom.
Okay.
We're taking
Comments.
1 and 5.
So which one?
Which one?
Fire, 5 liter, 2.5 liters.
Let's just do a 5 liter. Or do a 5 liter.
Yeah, go ahead and go to 2.
Correct.
That's the wrong lining. That's the wrong test one.
We're going for number 2.
That's one on the top guy on there. I'm gonna look up just a little bit. I'll keep you. Alright, that should be good. That doesn't look like it's fired. No, it look fired. Go ahead and hit fired. We're gonna fire 3 as well, just to be sure.
Okay.
We have switch right now.
Yeah.
Actually, we'll fire all of them. How about that? Just to make sure they're all working for the next time.
Yeah, that sounds good.
Jimbo,
I'm just gonna label these all as 16.
Yes, I agree.
So that's 2, 3, 4, and 6 fired.
Yes, that will be loaded.
Full stop at the end.
They were like 13 meters off bottom. And you fired all of them just to
Yeah, we fired
test?
all of them.
Gotcha.
So 5 was fired first, right?
5 was fired first, 1 was second.
Okay. So we fired 3, 4, and 6 just now.
2, 3, 4, and 6.
Okay.
We're not sure if 2 actually fires.
Right.
Okay.
I'm coming.
Keep that recording. Right? Or do you kill it?
All right, thanks everybody. We'll be back in a couple days. We have to transit.
Yeah, we've already ended it.
Okay, great.